As all of you come on in, I want to welcome you to the 2021 60th ACNP annual meeting. We're so happy to be here today in person. I wanted to welcome you and say that as you entered the room this morning, I hope you saw the memorial slides that have been portraying here. And I would like to have a moment of silence as we remember all of those dedicated scientists and colleagues that we have lost in the last year. Thank you. Welcome. Now it is my official duty or responsibility or pleasure to welcome you to the 2021 ACNP annual meeting. Welcome to our virtual attendees and to all of you that are here in person. I hope that you noticed as you walked in this morning, and that you can see here as well, that we are welcoming all of you. This is a slide, banner that the Diversity, Inclusion, and Equity Committee has created to welcome all of you of different races, backgrounds, genders, ethnicities to the annual meeting. We welcome all of you. As you know, this has been an exceptional year, and I can't say that without Sarah Tim and her wonderful team, this would not be happening today. So I want to thank Sarah Tim and Erin Shaw and all of her team that are here today. Thank you, thank you, thank you, Sarah. All right. So we're excited to have this in-person meeting, and I want to remind you of the health and safety reminders here. So all of the in-person attendees and accompanying guests, including your children, must be fully vaccinated to enter the convention center, and it's a requirement to upload your completed vaccination card during registration, and I'm sure you all know that already. Please wear your name badge at all times while you're in the convention center or other sponsored ACNP events. Please respect people's personal safety and precautions by the color of the lanyard that you're wearing today. Green is handshakes and high fives, yellow elbow bumps only, and red no contact. Keep your social distance. We welcome all of you, and we'll greet you in the way that you prefer to be greeted. I'd also like to remind you of the COVID code of conduct. So wear a face mask at all times unless you're actively eating, drinking, or presenting at a lecture or a panel. Poster presenters must wear a mask while presenting because we are limited in the amount of space that we have. Please practice good hygiene. Attendees that experience any symptoms or start to feel ill on site should quarantine in their guest room and contact ACNP's Executive Director, Sarah Tim. Please review the COVID Code of Conduct for returning home after the annual meeting and follow the guidelines. Now I would like to congratulate all of the 2021 travel awardees. Would all of you please stand? We are so excited about all of our travel awardees, and I want to especially thank the Education and Training Committee Chair, Bob Swift, and Vishali Bakshi for their work this year. Thank you so much, Bob and Vishali. And I also want to thank all of the members who volunteered to be our travel award mentors this year. Yay, thank you, everyone. So this year, I would like to recognize 3 of our named travel awardees. The first is the 2021 Harry June Travel Award. This award is available to early career researchers in substance abuse and was established in honor of our ACNP member, Harry June. Harry June Travel Award is funded by the National Institute for Alcoholism and Alcohol Abuse. Dr. June was accepted into membership in 2004, and he passed away in June of 2014. He was a renowned research scientist in the field of substance abuse, and he was a devoted mentor to students and young faculty. The 2021 Harry June Travel Award recipient this year is Sungwoo Kang, Assistant Professor, Augusta University Medical College of Georgia. Can you please stand? Right. The next is the Athena Marcoux Travel Award. It's available for early career researchers in neuropsychopharmacology. This award was established in honor of the ACNP fellow, Athena Marcoux, and her contributions to the field. To be eligible for this award, the applicant must have, through his or her research and teaching or clinical activities, demonstrated professional and scientific interest and achievement in the field of neuropsychopharmacology. This year we have two Athena Marcoux Travel Award recipients. The first is Catherine Moore, PhD, instructor at Johns Hopkins University School of Medicine. Will you please stand? And the second awardee is Monica Faulkner, who's a postdoctoral fellow at the National Institute of Drug Abuse. Could you please stand? Thank you very much, and congratulations to both of you. Would now like to welcome our ACNP advocacy affiliates. We would like to recognize them, and their contributions to the field are greatly appreciated, and we're so welcome to have them here at this meeting. The first is Diana Clark from the American Psychiatric Association. Diana, could you please stand? Here. The second is Laura Thompson, also from the American Psychiatric Association. Next is Jeffrey Borenstein from Brain and Behavioral Research Foundation. Again, welcome to the meeting. And then we have from the TARA organization, Treatment and Research Advancements for Borderline Personality Disorder Association, we have Valerie Poore and Sydney DeCaro. So at this time, it's my pleasure to turn the podium over to Maria Akendo, Chair of the Awards Committee, who will present the 2020 and 2021 honorific awards. Marina Wolf, who was Chair of the 2020 Awards Committee, is joining us virtually this morning.
Good morning, everyone, and thank you, Linda. Just going to let you know who was on the committee very quickly. So we had a wonderful committee and excellent discussions, and the committee members were Linda Brady, Cheryl Corcoran, Yogesh Dwivedi, Stephen Hyman, Lisa Monteja, Marina Wolf, and Carlos Zarate. Next slide, please. Next slide. Thank you. Okay, so the Daniel Efron Research Award is presented to an individual on the basis of outstanding basic research contributions to neuropsychopharmacology. The recipient of the Efron Award must be 15 years, within 15 years of their terminal training position as of December 31, 2021. The selection of the awardee is based on the quality of the contribution and its impact on advancing neuropsychopharmacology. The 2021 recipient is Kafui Jirasa, MD, PhD, Duke University Medical Center, and the 2020 recipient is Kay Tai of the Salk Institute for Biological Studies. Dr. Tai is arriving in Puerto Rico later this evening, and Dr. Jirasa will be accepting the award on her behalf. Please congratulate the recipients. Great. Yes, please come up to the podium. Thank you. Next slide, please. Thank you. Next is the Joe Elkes Research Award, which is presented to an individual on the basis of outstanding clinical research contributions to neuropsychopharmacology. The recipient of the Elkes Research Award must be within 15 years of their terminal training position as of December 31st of this year. The contribution may be based on a single discovery or a cumulative body of work. Of particular interest in selecting the awardee are contributions which further our understanding of self-regulatory processes as they affect mental function and behavior in disease and well-being. The 2021 recipient is Randy Auerbach of Columbia University, and the 2020 recipient is Romina Mizrahi of McGill Department of Psychiatry. They're both joining us virtually this morning, and please congratulate them for these accomplishments. Next slide, please. The Barbara Fish Memorial Award was established this year and is presented to an ACNP member who has made an outstanding contribution to basic, translational, or clinical neuroscience. The selection of the nominee is based on a mature body of work, notable for its impact, innovation, and depth. The 2021 recipient is Eric Nestler from Icahn School of Medicine at Mount Sinai, and Dr. Nestler is arriving in Puerto Rico later this afternoon. Please congratulate Dr. Nestler. Next slide, please. The Eva King Killam Research Award is presented to a scientist in recognition of an outstanding translational contribution to neuropsychopharmacology. The contribution should focus on translating advances from basic science to human investigations. The recipient of the Eva King Killam Research Award must be within 15 years of their terminal training position as of the end of this year. The 2021 recipient is Suzanne Amari from the University of Pittsburgh, and the 2020 recipient is Panos Roussos from the Icahn School of Medicine at Mount Sinai. Please congratulate them, and please come up to the podium. Next slide, please. The Julius Axel Award is presented to an ACNP member who has made an outstanding contribution to neuropsychopharmacology by mentoring and developing young scientists in the field. There were 2 award winners for both 2021 and 2020. The 2021 recipients are Peter Kalivas from Medical University of South Carolina and John Mann from Columbia University. Dr. Mann is joining us virtually this morning. Next slide, please. The 2020 recipients are Kathleen Brady from the Medical University of South Carolina and Blair Simpson from Columbia University. Dr. Brady and Dr. Simpson are both joining us virtually this morning. Please congratulate them. The Dolores Shockley Award for Diversity and Inclusion Advancement is given to an individual and/or program that has had outstanding success promoting diversity and inclusion within the fields of basic, clinical, or translational neuroscience. This award is formally known as the Dolores Shockley Minority Mentorship Award. Next slide. The 2021 recipient is Nii Adi from Yale University School of Medicine, and he is joining us this morning virtually. Please congratulate Dr. Adi. Next slide. The 2020 recipient is Greg Quirk from the University of Puerto Rico School of Medicine. Wonderful. Next slide, please. The Paul Hoke Award for Distinguished Service is presented by the ACNP to a member who has made unusually significant contributions to the college. The emphasis of this award is on service to the college, not for teaching, clinical, or research accomplishment. Next slide, please. The 2021 recipient is Marina Wolf, Oregon Health and Sciences University, and the 2020 recipient, next slide please, is Joe Coyle of Harvard Medical School. Dr. Wolf and Dr. Coyle are both joining us virtually this morning. Please congratulate them. Linda.
Just give me a moment here. Right. Again, I'd just like to briefly go over the code of conduct. Again, ACNP is dedicated to providing a safe and productive experience for all participants and attendees at the official meeting, regardless of sex, race, color, national origin, religion, age, physical or mental disability, perceived disability, ancestry, marital status, sexual orientation, gender identity, or any other basis protected by federal or pertinent state laws. ACNP does not tolerate discrimination, any form of prohibited harassment, and is committed to enforcing this code of conduct at its annual meeting and at any other ACNP event. Wanted to remind you of the photography policy. Meeting attendees should not take photographs or videos of any of the slides or posters or any presentation of data that are being presented. Thank you for allowing us to do this. This is what brings novel, refreshing, exciting new data to the meeting. All right, I'd now like to welcome you to the President's Plenary session. So today, the plenary is the BRAIN Initiative, Technologies to Understand Brain Function at Cellular, Circuit, and Systems Level. The 2021 President Plenary is going to highlight the development and application of BRAIN Initiative technologies that are enabling advances in understanding brain function at the single cell, circuit, and systems level in both animals and humans. The session is going to feature talks by Dr. Hangui Zhang from the Allen Brain Institute, Understanding Brain Cell Diversity, Dr. Justice Kupshaw from the Johns Hopkins University on sequencing the connectome, Dr. Damien Fair, University of Minnesota, Developmental Cognitive Neuroscience in the Era of Big Data, and Dr. David Boaz from Boston University, Imaging Human Brain Function with Functional Near-Infrared Spectroscopy in Ecologically Valid Settings. We're hoping that there's something here for all of you in this wonderful set of speakers. So our first speaker today is Hangui Zhang from the Allen Institute for Brain Science. Hangui is the Executive Vice President and Director of the Allen Institute for Brain Science. She received her PhD in molecular and cellular biology from Brandeis University, was a postdoctoral fellow at Massachusetts Institute of Technology. Since joining the Allen Institute in 2006, she's led several efforts to develop and operate high-throughput pipelines to generate large-scale, open-access databases and tools that accelerate neuroscience discovery. She's received many honors, including the 2016 Association for Women in Science Award for her research and interests in understanding neuronal diversity and connectivity in the mammalian brain, circuits, different cell types that work together, and how they process and transform information. At the Allen Institute for Brain Science, she has really built several really amazing research programs that we're going to hear about today. The Allen Mouse Brain Connectivity Atlas and the Human and Mouse Cell Types program to explore different properties of brain cells, and working towards the goal of creating a cell census and taxonomy of cells and understanding their connectivity in mammalian brains. Really excited to have Dr. Zhang here with us today. Thank you.
Good morning, everybody. First of all, I want to thank Linda for the invitation to this meeting and for the nice introduction. This is my first ACNP meeting. I look forward to interacting with many of you over the next couple of days. So today, I'm going to talk about our effort at the Allen Institute for Brain Science in trying to understand the diversity of cell types in the mammalian brain. And this work is also funded by the NIH Brain Initiative. The collective goal of neuroscience is to understand how the brain works. Can you see my cursor? Yeah, okay. This is a daunting task, given the extreme complexity of the brain itself, with millions to billions of neurons and a similar number of non-neuronal cells in the brain and the trillions of connections among all the cells. In order to tackle this complexity, it is essential to take a multi-level approach to understanding the organizing principles of the brain at all these different levels. Understanding the diversity of brain cells was set as the first top priority of the Brain Initiative, because it's really the foundation, it lays the foundation for us to understand how brain works at the exquisite detailed level. So why do we need to study cell types and how to do that? To understand any complex system, such as the brain, an essential first step is to create a parts list of that system. And for the brain, we can consider a catalog of cell types as that parts list. With the parts list in hand, we can then also create consistent genetic access of the cell types to allow us to do observation and manipulation in a reproducible manner. The process of understanding cell types will allow us to discover not only the new cell types, but also the new relationships across different cell types. It will also help us to understand a lot of the other important biological processes, such as development, evolution, brain circuit, and function. And finally, of course, having a detailed list and understanding of the cell types will also help us to better understand diseases, a number of different diseases, the differential vulnerability of the different cell types under a variety of diseased conditions. So with all this importance, how do we generate a comprehensive list of cell types? We think that there are three essential requirements. The first of all, we need to take a systematic approach, striving for completeness in characterizing the cells at the individual single cell level and covering the entire nervous system, in order to discover most, if not all, the cell types that comprise the brain. And secondly, we should take a systematic and standardized approach, perform as many quantitative measurements as possible, and use a data-driven approach to achieve an unbiased categorization of cell types that allows comparison of the different cells in different systems in a more objective manner. And finally, because cells exhibit diverse properties, it is important that we look at cells, profiling them at different levels, for example, at molecular gene expression level, at physiological functional level, and at morphological and connectional level. And looking for congruency, consistency of properties across these different levels to achieve what we call an integrated definition of cell types that can be more widely applicable. So that the findings from one lab can be easily related to investigations in another lab. Otherwise, if every time you use a different method to look at the cells, different types, you get a different answer, then the concept of cell types becomes not that meaningful anymore. So with this kind of a motivation, at the Allen Institute, we have set up multiple technology platforms to investigate individual cells and their different properties, and in particular, associate the cell type properties with their connectivity patterns. Our front-end approach is to use this highly scalable single-cell RNA sequencing method to profile tens of thousands to even millions of individual cells, and using the transcriptomic profiles of the individual cells to perform a systematic clustering analysis and classification to come up with this comprehensive taxonomy of cell types. We also perform single-cell epigenomic profiling, such as the single-cell ATAC-seq, and combine that information together with the transcriptomics to define gene regulatory landscapes at the individual cell level. By using the molecular information gained from this kind of large-scale single-cell sequencing approach, we also create genetic tools to target individual cell types, and that would include transgenic driver lines, as well as now, in more recent years, potentially using enhancer elements discovered in the genome to create viral vectors that can target different cells in a more flexible manner. We use spatially resolved transcriptomics to profile not only the the distributions and relative proportions of the individual cell types, but also using this as a post-hoc approach in conjunction with anatomical and physiological profilings of the cells to decode the molecular identities of cells that, for which we have obtained connectional and functional information. We also use a multi-model approach called patch-seq, from which to obtain multi-model information from the same cells. And that includes the intrinsic electrophysiological properties through patch clamp recordings, the local dendritic and axonal morphology information from the recorded cells, and finally, the whole transcriptomes from the cellular content extracted from the individual cells. And by obtaining this kind of a triple modality information, we can then perform clustering analysis and define cell types using all the different types of information together. For long-range projection neurons, we also try to link their molecular profiles with their long-range projection patterns by reconstructing the full brain-wide morphologies of individual cells that are defined genetically. We have a large-scale electron microscopy pipeline to generate high-resolution, nanometer-resolution electron microscopy data sets from particular brain regions, such as the visual cortex. And from that, we obtain synaptic information from individual cells that are defined by their morphological patterns. In more recent years, we also are beginning to incorporate other advanced new technologies such as barcoded connectomics, for which you will hear a lot more from the next speaker, Eustace, in which we use the barcoded viruses that will allow us again, to link molecular identities of the individual cells with their long-range projection patterns in a highly scalable manner. And finally, we're establishing large-scale light microscopy-based approach to look at the brain structure and connectivity, as well as linking that with the protein expression patterns in large brain volumes. Behind all these large-scale experimental platforms, we strive to establish also very strong informatics, data science, and technology platforms that will allow us to really manage all these different large-scale data types from the different pipelines that generated from the different pipelines that will allow us to perform computational analysis of the data and finally organize the data and the knowledge and present them in the public, in open public databases. That's a unique characteristics of the work at the Allen Institute. So with all these different technology platforms, now I will tell you a little bit about the work that have been done at the Allen Institute in characterizing cell types in different parts of the brain. Our starting point is the cortex. And here is just a basic introduction of the major neuronal types found in the cortex. The first major type is the glutamatergic excitatory neuron types that can be excitatory neuron class that can be divided into several major subclasses. As you know, the glutamatergic neurons in the cortex are nearly all long-range projection neurons. They have widespread projection patterns both either within the cortex or going out of the cortex into many subcortical regions. So the largest subclass of excitatory neurons is the intratelencephalic, or IT, neurons that are distributed across all 6 layers of the cortex. And they can be grouped into different types based on their lamina specificity. The second major subclass is the extratelencephalic, ET, or pyramidal tract, PT neurons that are mainly localized in layer 5, that are the major output neurons from the cortex. They have projections into a large number of subcortical targets. The next major subclass is the corticothalamic neurons that are mainly localized in layer 6, and their primary targets are in the thalamus. In addition to those 3 major subclasses of excitatory neurons, through our transcriptomic studies, we also identified 2 other major subclasses. One is called the layer 5 MP, that are near projecting, and the last one is layer 6B, subplate neurons. The other major class of neurons in the cortex is the GABAergic inhibitory neurons that have mainly local projections within the cortex. The GABAergic neurons exhibit extremely complex electrophysiological and morphological diversity, and they can be grouped into many different types based on their morphoelectrical properties, as well as based on a number of neurochemical marker genes that have been well identified and characterized in the field. So, as I said, our starting point of characterizing cell types is using the single-cell sequencing approach. We mainly have been taking 2 different sequencing methods. The first is the full-length deep sequencing SMART-seq approach that gives us high-density transcriptomes of individual profiled cells. The scalability of this method is relatively limited. The second approach is the droplet-based 3' end sequencing, shallow sequencing method that is highly multiplexible, and we have been using the 10x genomics platform, 10x Chromium, to do that. In the first set of published studies, we investigated a couple of two different brain cortical regions from the mouse, and that's primary visual cortex and anterolateral motor cortex, and one cortical region from the human brain, the medial temporal gyrus, using the SMART-seq approach. By profiling tens of thousands of individual cells, we are able to classify the cortical cells, both neurons and non-neurons, into different types. And what we found that the cortical cell types can be organized in this hierarchical manner, that is highly conserved between mouse and human. The hierarchical structure of the cell types are very well conserved, along with the major subclasses and types of cells as shown as the major branches of the hierarchical tree. At the same time, we also identified species differences, and those differences are mainly found to be present at the terminal leaf level, the individual clusters of the transcriptomics, as well as the differential expression of the individual genes, even at the major branch level. So that's a cross-species comparison. We find both conservation and species differential properties. What about cell types across different cortical regions? How do they look like? Are they similar or different? So in this mouse cortical study, in which we investigated two different cortical regions at the opposite end of the cortex, we found, interestingly, this dramatic dichotomy of the two different major neuronal classes. We found that the GABAergic inhibitory neurons are well conserved or shared across those two different cortical areas, and that's shown by both the contribution of cells from both regions into the same transcriptomic clusters, as well as the very low number of genes that are differentially expressed between those two different regions. The excitatory glutamatergic neurons, on the other hand, are highly distinct between those two different regions, and this is shown by the almost exclusive contribution of cells from one of the two regions into each of the transcriptomic clusters, as well as the high number of differentially expressed genes found in the homologous pair of the transcriptomic clusters across those two different regions. So then beyond the transcriptomic level, we wanted to understand what do those transcriptomic cell types mean? How do they correlate with the commonly recognized morphological and physiological properties of the individual cells? And for this, we profiled a large number of GABAergic interneurons in the mouse visual cortex using the patch-seq approach. And in this particular study, we obtained transcriptomic as well as electrophysiological information from over 4,000 individual patched neurons. And from those 4,000 neurons, we reconstructed the local morphologies from 500 of those neurons. So in order to investigate the relationship between transcriptomics and morphoelectrical properties, we performed independent clustering just using the extracted quantitative morphoelectrical properties of the 500 neurons and compared their ME type identities with their transcriptomic or T-type identities. And as shown in this cross-correlation diagram, because every cell has both the T-type and ME-type identities, we see a high degree of concordance, a very high degree of concordance across all the major types between transcriptomic identities and morphoelectrical identities. At the same time, you can also see that this correspondence is high, but not perfect. We rarely see one-to-one correspondence between those two different classification schemes. Very often, we see that the correspondence is several to several. So indicating a differential phenotypic variation at more refined levels. So in order to achieve a better, more consistent definition of cell types across all these different modalities, we used the mapping statistics in both types of clustering to derive at what we call the MET types, or triple modality integrated types. Within each of those MET types, the consistency of the properties of the individual cells becomes a lot higher. So using this approach, we defined 28 different MET types for all the GABAergic neurons from the visual cortex. And those 28 MET types are grouped into very well-defined major subclasses of the cortical interneurons, and that include the parvalbumin positive and somatostatin positive interneurons that belong to the MGE class, as well as the LAMP5 positive neuroglialiform cells, the VIP positive cells, as well as the SNCG positive multipolar interneurons that are mostly coming from the CGE developmental origins. So the major conclusion of this PATCH-seq study is that we find that the integrated MET types have intermediate granularity. We found only 28 of them instead of 60 or 40 when we use different modalities to classify the cells. Despite of the intermediate, or maybe because of the intermediate granularity, we find a high cross-modality concordance as well as high mutual predictability between the different individual cellular properties. The MET types also recapitulate traditionally defined interneuron types very well, as shown in the labels using conventional names of the different GABAergic interneuron types. And we also find a further subdivision of those classically defined interneuron types. And those further subdivisions defined by transcriptomics are usually driven by layer specificity. For example, for the many different kinds of basket cells that we find in the cortex, the transcriptomics would group the different basket cells based on their layer specificity from superficial to middle to the deep layers. Similarly, for the many different kinds of somatostatin positive cells, we are also able to group them into Martinati or non-Martinati-like across the entire cortical depths, and that's mainly driven by layer specificity. Finally, the definition of the MET types then allow us to establish a set of Rosetta Stones that can bridge molecular identities and other properties of the cells. And this then can allow us to infer the different cellular properties from very different data types. For example, we use this PATCH-seq information now, then we're able to identify molecularly defined cell types from our electron microscopy data set, in which cells can only be defined using their local morphological features. But then, through this kind of Rosetta Stone kind of information, we can then assign molecular labels to cells with synaptic connectional properties in our EM data set. Now, as Linda said, we are participating in the NIH Brain Initiative effort now with several large grants from NIH. We are part of this consortium called NIH Brain Initiative Cell Census Network, BICCN, in which many investigators across the country, as well as several other countries in the world, using a very similar set of technology platforms to profile cells in multiple mammalian species, from mouse to non-human primate, such as marmoset and macaque, to the human brain itself. And by integrating all this different multi-model information together, we wish to obtain molecular taxonomies that are consistent across different mammalian species. And we want to obtain spatially resolved molecular taxonomies using a combination of sequencing approach and spatial transcriptomics approach. We want to define cell types by integrating different modalities of information together, as I mentioned, using the patch-seq approach. And finally, we want to derive cell type-based input-output wiring diagrams across the different brain regions. The first landmark achievement of BICCN, in which I'm particularly proud of here, is the publication of 17 research articles in a special BICCN issue in Nature that appeared in October, early October this year, which really showcases a variety of highly detailed characterization of cell types across multiple mammalian species at multiple levels, molecular, transcriptomic, epigenomic, physiology, morphology, and connectional. And we are doing this work in a number of different brain regions now. The centerpiece of this collection of work is another large-scale multi-model characterization of cell types in another major cortical region, primary motor cortex, that was characterized in mouse, marmoset, and human. And through this large-scale consortium collaborative effort, we obtained this taxonomy or atlas that's anchored to the highly detailed mouse consensus transcriptomic taxonomy, but then also linking the transcriptomic and epigenomic information from multiple species, morphological and physiological and connectional information and that are characterized in detail using many different methods in mouse. The major conclusion from this collaborative effort is, again, really revealing several major organizing principles of the cell types in the cortex. And that is summarized here. First of all, again, the cell types are organized in this hierarchical manner. We find the coexistence of discrete and continuous variations of different phenotypes. And the discrete variations are mainly residing at the major branch level, whereas when you go into the more refined terminal leaf level, the phenotypic variations become a lot more continuous. We also observed concordant and differential phenotypic variations across different modalities. Again, the concordance at the major branch level, whereas you observe more differential variations at a more refined level. And finally, cross-species conservation and divergence follows the same kind of rule. Higher degree of conservation at a major branch level, whereas more divergence at both individual gene expression level and at a cluster level at a more refined branching level as well, suggesting that the understanding and classification of cell types, you really need to take this kind of a holistic view and define cell types at the proper granular level. So, what I have told you about is really mainly the studies of individual cortical regions. What happens if you look at beyond just a particular regions? When you examine the cell types across multiple brain regions, will you be able to uncover the relationships of cell types across those different regions as well? Okay, so before I go into that, I also wanted to highlight a particular study here, which is a combination of transcriptomic and epigenomic information together that allows us to define cell type specific gene regulatory landscapes, and which will also lead to the ability to generate cell type specific targeting tools. So in this particular study, we, in collaboration with multiple groups from the Salk Institute, UCSD, and Broad, we obtained many different data sets from both single-cell RNA sequencing and single-cell epigenomic sequencing that covers both chromatin accessibility and this DNA methylation patterns to generate this integrated taxonomy that utilizes all the different single-cell information together. We can see that this integrated taxonomy is able to integrate the different disparate data sets very well, which then allow us to combine the transcriptomic and epigenetic genetic landscapes together. For example, for this particular gene, we are able to identify cell type specific gene expression patterns, as well as associated epigenetic peaks that are shown by both the ataxic peaks as well as the methylation peaks, and then identify cell type specific enhancer elements that then allow us to develop viral targeting tools to target the major subclasses and types of cells in the cortex for both excitatory neuron types as well as inhibitory neuron types. And we also further show that the enhancer-based viral vectors can work well across species, not only working in mouse, but then they can also be applied to non-human primate brains, as well as human ex vivo brain slices. So, okay, now move on to the multi-region cross-area comparative studies. In this another much larger scale single-cell transcriptomic study, we profiled over 1.3 million cells across the entire mouse cortical sheet. So in this study, we covered all the sensory, motor, as well as associational cortical areas of the mouse neocortex or isocortex, as well as a evolutionarily similar but more ancient structure, hippocampal formation. For the hippocampal formation, also called archicortex, in this study, we covered not only the hippocampus itself, but also the nearby subicular complex, multiple regions of the subicular complex, as well as the medial and lateral entorhinal cortex. By combining this large-scale transcriptomic data together, which used both 10x shallow sequencing approach and the SMART-seq deep sequencing approach, we come up with this much larger transcriptomic taxonomy with a much extended number of cell types. So, and we then also used the many gene markers identified from this transcriptomic taxonomy to do extensive and detailed anatomical annotation of all the clusters, and we are able to assign the anatomical locations, including both regional specificity and layer specificity to all the individual clusters, and again, group them in this kind of a hierarchical manner, from neighborhoods to subclasses to supertypes to the individual clusters or types. And what we found is the previous rules that we identified from the study of 2 individual cortical regions still apply to this much larger data set, but also at the same time, the rules themselves have also evolved with the new information gained from this study. First of all, on the GABAergic side, in the GABAergic neuronal class, we find that a vast majority of the individual GABAergic types are still shared across multiple cortical areas, as shown in this kind of a rainbow-colored regional distribution patterns. At the same time, as you can see here, we also identified a number of highly distinct GABAergic interneuron types that are particularly specific to certain regions, and those certain regions are all shown in this kind of greenish color, which indicates that they come from the hippocampal formation regions. So in addition to all the shared GABAergic types, we also now identified a set of highly distinct GABAergic types that are specific to hippocampus and its neighboring hippocampal formation regions. On the other hand, the glutamatergic class of neurons, the diversity has expanded dramatically. We see not only highly distinct cell types in specific regions, in particular, the distinction between the green-colored hippocampal formation regions and other neocortical or isocortical regions. We also begin to share a lot of see a lot of the types that are shared between neighboring cortical areas. So in the next couple of slides, I will unpack the complexity of the glutamatergic cell types a little bit further. So here, we want to first look at the distinction and similarity of the excitatory neuron types between isocortex and hippocampal formation. We found that through this UMAP display of the different transcriptomic types that are colored either by regional specificity or subclass or cluster specificity. The cluster is shown by this constellation diagram that's modeled after the UMAPs. We found that the major continents of this UMAP comprises of cell types from the isocortical regions that includes all the major subclasses that I told you early on, that include the intratelencephatic neurons across different layers, from layer 2-3 to 4-5 to layer 6, the highly distinct layer 5 PT, subcortical projecting cells, as well as the cortical-thalamic projecting layer 6 cells and its similar closely related MP and layer 6B cells. And many of the cells in the isocortex are actually now found in this kind of shared manner across the different regions, and which we found to be distributed in a gradient manner across the cortical sheet. Besides those cell types from the isocortical regions, we also identified a number of islands, highly distinct islands of cell types that are all coming from the hippocampal formation areas. And through a molecular homology search approach, we find that all the different cell types coming from the hippocampal formation actually have high degree of molecular similarity with the major subclasses of the cells from the cortex. And to confirm that molecular similarity, we identified the marker genes and looked at the laminar distribution of the marker genes for the hippocampal regions. So we find that they also exhibit a similar layer or laminar distribution, confirming the homologous relationship between the cell types coming from different hippocampal formation regions such as entorhinal cortex and para-post-pre-subicular regions. And furthermore, we find a very interesting similarity between the major output cell types from neocortex and from hippocampal formation. The major output neuron subclass in the cortex is this layer 5 PT or extratelencephalic neurons. We find very few homologs, except for a single cluster from the layered structure of the hippocampal formation, including entorhinal cortex or different subicular para-post-pre-subicular areas. However, we found very strong homology or similarity with the neurons from the CA1 and subiculum itself. Neurons from CA1 and subiculum in the hippocampal formation are known to be the major output neurons from the hippocampal formation. So again, a molecular homology between those two different regions is related to the anatomical or connectional property between those two different regions as well. So to make a long story short, here is a diagram summarizing all the different glutamatergic excitatory neuron types that we identified from those two major brain structures, the isocortex and hippocampal formation. The 240 also clusters in those two different regions are organized in a region-specific and layer-specific manner that are highly intersected with each other. The left side is all the cell types from isocortex that are divided by regions, and the right side are all cell types identified hippocampal formation. They are also organized in a layer-specific manner from superficial to deep, with the major projection types, intrathalamus-cephalic, superficial here, and deep layers here. Extrathalamus effect, the output long-range output projection neurons in both regions are here, and then the cortical-thalamic specific projection cells are shown here. So a highly parallel cell type organization between two major brain structures, cortex and hippocampal formation, with all the major subclasses of long-range projecting excitatory cells preserved in both structures, suggesting a high degree of evolutionary relatedness between those very different brain regions, and also now forming hypothesis to us that those different major subclasses cells may be having similar long-range connectional or projectional patterns as well. Finally, as you can see from this highly complex molecular structure, within the isocortex or neocortex itself, we see a combination of region-specific cell types, in particular cell types coming from different associational cortical areas, such as anterior cingulate and retrosplenial cortex, in combination with a gradient distribution of many different kinds of cell types that are shared across those two regions, the many different regions. So the combination of regional specificity and a gradient distribution of shared cell types collectively defines the regional specificity of the different cortical regions, as well as the modularity, the different modules that are of the cortex that we believe form the molecular basis of the cortical modularity that have been observed by connectional and functional properties across different mammalian species, from mouse to non-human cortex, non-human primate to the human brain itself. So, okay, so then in the final set of study, I want to further illustrate our attempt in linking molecular identities with the connectional patterns of the individual cells. And this is by looking at a large number of long-range projecting neurons across different regions using the full morphology reconstruction approach from genetically defined cell types. For this particular study, in collaboration with a group in several different groups in China, we established a pipeline for single neuron morphological reconstruction that combines genetic sparse labeling with high-resolution full-brain fluorescent imaging with computationally derived reconstruction, registration of the whole brain data set, to finally data-driven quantification and classification approach. I want to use a particular example to illustrate the morphological diversity that we have observed in transcriptomically distinct or even similar cell types that we identified. And in this particular example, we focus on a particular type of cells in the cortex that's called the CAR3, defined by this gene called CAR3. So the CAR3 subclass of cell types in the cortex is shown in this UMAP. Is a highly distinct subclass by itself. It's comprised of cells coming from a number of cortical regions, mainly located in the lateral part of the cortex from across the entire anterior-posterior span of the cortex, as well as cells coming from this small region called the claustrum that's sitting underneath the agranular insular cortex. So the cells belonging to the CAR3 type comes from many different parts of the cortex, as well as the claustrum itself. And the cells exhibit highly homogeneous transcriptomic properties. We reconstructed about 100 individual cells from this particular type, and we observed extreme morphological or projectional diversity. All the cells have, all 100 cells belonging to this transcriptomic type have only intracortical or cortical-specific projections. And through this, and from this matrix, projection matrix, in which each cell is shown in one column, and each of the target region is shown in a row, we see that the projection patterns of each of those 100 cells are very highly diverse and distinct from each other. We are able to cluster their projection patterns into 13 different types, and the major division between those two, all those clusters is the division between claustrum neurons and cortical neurons. But even within each of those major divisions, we observe tremendous diversity at the individual cluster level and at the individual cells level. And we find that this projection diversity is really driven by a topographic rule, meaning that the cells coming from with their soma located in different parts of the cortex, from anterior to posterior, would have a very distinct projection pattern based on where that cell is located. The cells located in the more anterior part of the cortex would have projections in a particular way. For the cortex, it's mostly in the nearby regions, in the anterior part of the other cortical regions, and the cells located in the posterior part would have a distinct patterns that are consistent with their local locations as well. So beyond that particular example of CAR3 neurons in the cortex and claustrum, we also examined altogether 11 major projection neuron types in cortex, thalamus, and striatum, and we see a consistent kind of diversity patterns across those two major, across all those major types. First of all, we see highly distinct projection patterns among all those major 11 molecular cell types. But in addition to that, within each of those molecularly defined cell types, we also still observe a high degree of diversity, and that diversity of long-range projection patterns is mainly reflected at 3 levels: regional specificity, topographical specificity, as well as projection variations at the individual cell level. Every cell chooses a subset of the projection targets that this, its entire population or type have. So finally, I think I'm running out of time now. Finally, I wanted to say, going beyond all these different multimodal studies, we under the BRAIN Initiative are marching towards our ultimate goal of generating a taxonomy of cell types across the entire brain. And the first step of that is to generate a classification scheme for the cell types across the mouse brain. And here is just one preliminary data set showing that we have expanded our transcriptomic work to cover the entire forebrain of the mouse. That includes regions, not only the cortex, hippocampal formation, but also other regions such as the striatum, amygdala, thalamus, and hypothalamus. And from this tremendous data set, which now contains nearly 3 million individual cells, we are able to obtain a transcriptomic taxonomy that contains more than 2,000 individual clusters. And we are very excited to see the variety of patterns of relationships, again, across the different clusters, and we are eager to annotate this data set and again derive rules that governing the diversity of cell types within each of those regions, as well as the relationships across the different regions. So with that, I want to summarize our work by highlighting the following main points. First, I hope I have shown that the large-scale single-cell transcriptomics can provide a framework for cell type classification and defining the diversity of cell types across different brain regions. We found that the cortical transcriptomic taxonomy reveals a hierarchical organization of the different cell types, the coexistence of discrete and continuous variations, and similarity and difference across different species. We found that a combination of single-cell transcriptomic and epigenetic profiling enables the discovery of enhancers and the potential creation of novel cell type targeting tools. I showed that a multi-model classification refines a set of cortical interneuron, MET types, that can give us higher degree of concordant transcriptomic, morphological, and physiological properties, and higher degree of mutual predictability across those different properties. From multi-regional combined transcriptomic studies, we identified many highly distinct excitatory projection neuron types that are present in different parts of the cortex and hippocampus. We also find gradient distributions of those types across different cortical areas, as well as gradual but discrete transitions between neocortical and retrohippocampal regions, reflecting their potential evolutionary relationships. And from there, we think that multiple parallel neural networks can be formed by homologous sets of transcriptomic types that are obvious, within cortex and hippocampus, but maybe going beyond that and maybe present in other brain regions as well. We further find that complete brain-wide single neuron morphology reconstruction studies reveal distinct morphological and projectional properties not only between major transcriptomic types, but further extensive diversity existing even within relatively homogeneous transcriptomic types. And finally, I hope I have shown that this kind of comprehensive multi-model characterization of cell types demonstrated that all these different cellular properties can be integrated together, bridging the analysis across the different modalities, as well as relating cellular properties across different species. So with that, finally, I want to thank a large team of scientists at the Allen Institute who have contributed to this systematic cell type characterization effort. I want to thank our founder, Paul G. Allen, for his generous support, as well as many different grants that we have obtained in different NIH institutions, both within Brain Initiative and beyond. And finally, our collaborators within the BICCN consortium, among the multiple data modalities and technology platforms that I presented to you today. So with that, thanks for your attention.
Great. Thank you so very much, Dr. Zheng. In the interest of time, we'll hold questions to the end, and I'm hoping that we will be able to do that. Otherwise, you can come up and ask questions of the speakers or follow up with them during the meeting itself. So our next speaker is Justice Kebshol, who is an assistant professor at the Johns Hopkins University. Dr. Kebshol grew up in Germany and received his Master's of Science from the University of Cambridge in UK in 2011. He pursued his PhD in Tony Zadar's lab at Cold Spring Harbor Lab, where he developed DNA sequencing-based tools for brain mapping. In 2017, he joined Lingqing Guo's lab at Stanford University as a postdoctoral fellow, and he was working on the evolution of cerebellar nuclei before starting his own lab at Johns Hopkins. His research focuses on how brain areas, circuits, and cell types change across evolution to produce complex network architecture. His lab engineers cutting-edge circuit tracing and viral tools to map brain circuits and networks at very high resolution in a variety of species, including mice and humans, as well as birds, reptiles, and amphibians, so that he can compare and contrast across species. This will be a really interesting talk that nicely follows the one from Dr. Zhang. So, welcome.
All right. Good morning, everybody. Well, first of all, thank you for the kind introduction and the opportunity to speak here today. Okay. Well, as you've just heard in this beautiful talk by Honghui, single-cell transcriptomics has really proven to be an incredibly powerful hammer to answer all kinds of questions in basic neuroscience, but also in translational and clinical applications. But as Honghui has also beautifully shown, neurons vary in much more than just which genes they transcribe in the adult state. They vary in their morphology, in their electrophysiology, and critically, I think, in their connectivity. And so in this talk, I want to really focus on connectivity and introduce a few of the methods that we've been developing to try to add single-cell resolution connectivity information at scale to these transcriptomic atlases that we're building. Now, first off, what are the issues here? Why are we not already swimming in a sea of single-cell connectomic data? I think the first one and the biggest one is what I call a mismatch of scales. And this is a problem that is common to all of neuroanatomy. This is the problem that neurons are microscopic structures, right, like single cells. The axons are thin, synapses are tiny, but nevertheless, they span these macroscopic distances, axons stretching from one hemisphere of cortex to the other, from motor cortex to the spinal cord, centimeters, meters, right? And so this makes it, makes brain mapping really annoying and forces generally a trade-off in what kind of resolution we can map the brain at what speed, and whether we get single-cell resolution or not. What that looks like in practice is something like this. So our workhorse in connectivity mapping, as you all know, is viral-based projection tracing, where you inject your favorite GFP, say, encoding virus into your area of interest. All the cells light up, right? The axons get filled with GFP, and then you can image the whole brain. And wherever you see green axons, you know that your area is projecting to. Now, that is critical and foundational information upon which we can build. But similar to bulk RNA sequencing, to single-cell sequencing, what we're doing in these bulk tracing approaches is average over all the cells that we've labeled in the same color, right? And so just given bulk connectivity information, we cannot distinguish between fundamentally different modes of wiring. For example, in this example here, where you have one source region projecting 3 downstream target areas, if you just trace at bulk resolution, you cannot distinguish the possibilities that maybe there's 3 dedicated populations of neurons, each one specialized to project to just one downstream target area, or maybe alternatively, there's neurons that project to all the downstream target areas, really just broadcasting information. Now, that's completely different implications about how, which computations can be happening in this circuit, and obviously we'd like to know. Now, if you want to do this, classically what you have to do is to actually trace single cells one at a time, and what you do is you take the same tracers you use for bulk tracing, just inject very, very little of it, so that by chance you just label a single cell per brain. Now, that cell lights up, right? You get beautiful images like this once you image the whole brain, but it's very tedious. It takes a long time. You only get one cell per animal. People in the Allen Institute, for example, and at Genelia and the Mouse Light project have been working really hard to try to multiplex this to trace more than one cell per brain. And generally, the strategy for this is to find ways to sparsely label cells so that they're somewhat separated from each other inside the brain, and then image the brain volume at very, very high resolution so that it can actually follow individual axons throughout the space. That works, right, and gives us lots more single-cell tracing data than we ever had before. But it comes at the cost of having to do this really high-resolution imaging, which is expensive and slow still. And also, even with the highest resolution tracing that we have right now, we're still limited to mapping out maybe a few dozen cells per brain region per animal. If you want to, you might think you can multiplex this further by using different independent color channels. So, obviously, if you trace a red neuron and a green neuron and a blue neuron, you get 3 times as many neurons per brain as before. But the problem using colors is that we really aren't very good at putting very many different colors into the brain and then reading them out reliably again. So that we're usually stuck to 4 to maybe 10 different colors inside the brain. And while that is better than just one color, if you think about the complexity of the brain, with a mouse brain having 10 to the 8 neurons, even like a regular area like the mouse auditory cortex having 300,000 or so neurons and over 100 cell types, understanding diversity of neuronal projections, one or even a dozen cells at a time, is going to keep us busy for a long time. So that's a mismatch of scales. But even if we overcame this problem of brain mapping itself, if we want to integrate our connectivity information with the transcriptomic information that we can get by single-cell sequencing, for example, we're still facing a hurdle, which is that generally these very high-resolution imaging approaches are not very good for allowing us to also measure gene expression data. And so we can trade off and either get good gene expression or good tracing, but both at the same time is challenging. Now, I guess almost a decade ago now, Tony Zeder from Cold Spring Harbor, my old advisor, introduced this idea of sequencing the connectome, incidentally the talk title. And his idea was, why are we trying so hard to use different colors and high-resolution imaging to map single-cell connectivity if ultimately colors are just not diverse enough to map all the cells in the mouse brain, and we will always fall short. Why don't we use a different label that actually has enough diversity to trace not a few cells per brain region, but maybe eventually all the cells in the mouse brain at the same time? And the label that he came up with is cellular barcodes, random nucleic acid sequences. And this is an idea borrowed from developmental biology, where people have been using sequences to label and track cells over time for a while now. And the beauty of using barcodes is that because they are linear sequences of nucleotides, they have a huge combinatorial diversity. So at each position in the sequence, you have 4 possible choices of bases, A, T, G, or C. And so if you just string those together in a little barcode, you build up diversity exponentially. So with 30 random nucleotides, you have 4 to the 30 or 10 to the 18 possible different sequences available, 10 to the 18 different labels, which means you have 10 billion times more labels than you would need to label every single neuron in the mouse brain uniquely. Now, critically, we can really quickly read out and reliably read out these sequences, these barcodes, by Illumina sequencing, and they are in the same modality fundamentally as the cell's transcriptome because the mRNA is already DNA. And so by switching labeling strategies from using fluorophores and imaging to using barcodes and sequencing, I think we can actually overcome these two issues, the issue of scale or mismatch of scales and the issue of compatibility between connectomic and transcriptomic information in one swoop. Now that left us with the question of how you actually use barcodes to map on report connectivity. And so in this talk, I want to give you an overview over what we've been up to in this last decade and the methods we've been developing to try to get to single-cell resolution at scale. And so I'll first be talking about MapSeq, the first method we've developed in this line of work. Then I'll speak about how we have been trying to bring high spatial resolution back into this barcode-based connectomics using in situ sequencing approaches. And then I'll quickly touch on multi-omic readouts, where we actually tried to combine transcriptomics and connectomics for the same cells. And I'll finish with how we're scaling these approaches to the whole brain now. So first off, MapSeq. So MapSeq is the first method we developed, and it's essentially allowing you to do massively multiplexed single-cell projection tracing. So basically, we do this, but for thousands or tens of thousands of cells in the same brain and in just a single experiment. So how do we go about doing this? So the first step is to make these diverse libraries of viruses. So it's a pool of different viruses where each virus particle essentially encodes a different barcode sequence. We can now make like 100 million different diverse barcode libraries. Then you take this highly diverse library and inject it into your favorite brain region. And you inject it at a concentration such that each neuron that gets infected on average with just a single virus particle. And then that neuron is uniquely labeled by the barcode carried by the virus particle, and we can express that barcode as an mRNA and prop the cell full with this mRNA just as we would usually fill it up with GFP protein. In a belts and suspenders approach, then we co-express a engineered presynaptic protein that specifically clamps onto and binds to the barcode mRNA and drags it along when the protein gets trafficked by the cell into the axonal processes. And so what you end up with then is a brain with thousands of cells uniquely labeled with different barcode sequences, and all the axons coursing through the brain being stuffed full with these barcode mRNAs, each molecule of which, again, is encoding the barcode identity and therefore which cell it's coming from. So if you then take the brain and chop it up into little pieces and sequence all of them to determine which barcodes you find where, you can reconstruct the projections of these single cells all in one go. And what this looks like is, say, if you look up here in this brain region, if you find barcode 78 there, what that means is that there's a neuron labeled with barcode 78 that is sending projections up into this top corner here. If you then look in a different brain region and you also find barcode 78 there, then that means that that same neuron is projecting both up here and down here. If conversely you find barcode 43 only down here, that means a different neuron labeled with barcode 43 is projecting only into this region down here and not up there. And so just by going through the list of all the thousands of barcodes you have, you can reconstruct these single cells connectivity or projection matrices. What this looks like in real life is something like this, where we've traced out over 6,000 cells in the from the mouse auditory cortex, projecting to a few of their brain-wide targets in just a couple animals. So we've done our homework and done all the controls to try to figure out if this is actually faithfully mapping single-cell projections. And over our different studies, we've always found that MapSeq works as efficiently as traditional retrograde tracers, CTB, or retrobeats in detecting any given projection target, and we see no evidence of distance-dependent filling. And actually, if we take our MapSeq neurons and compare them to neurons traced out the traditional way using fluorescence, we can actually not distinguish them at a statistical level. So that made us feel pretty good about the method. Well, anyway, so that's the design of the method, but what kind of questions can you actually answer using these tools? And so the story I want to tell here is one where we try to ask, how does information flow through cortex? How do different cortical areas communicate with each other? How does intracortical information flow happen? And so we did this in collaboration with Tom Mercer-Klogel in London and Yunyun Han and Rob Campbell in this lab. And so when we started the study, one model of intracortical communication that was kicking around the literature was that this one neuron, one target model. That the idea that there would be specialized neurons that projected just one downstream target region, and so would therefore be, have the ability to ideally tailor the output to the needs of that target region. Now that's easy to think about and very attractive as a model, but when we actually looked at the tracing data that was available and out there, that was actually not the only one that was possible. Indeed, it was possible that individual neurons project largely to more than one target area, broadcast out to multiple targets, and they might then do so either by randomly choosing these downstream target areas or actually acting as what we would call projectional cell types, where some regions are always co-innervated and others never. Now, to figure this out, we looked at the primary visual cortex in the mouse and looked at its projections to the higher visual areas right surrounding it. And we started this using actually doing standard single-cell fluorescence tracing. And so we patched onto single V1 neurons, filled them, electroporated the GFP construct, and then filled those neurons and imaged the whole brain, and got images like this, where already in this one, you can tell that this neuron doesn't project to just a single cortical area, but actually has projections to more than one. When we quantified all the cells that we traced, we indeed found that over 70% of all neurons did project to more than one target area, and often even up to 5 or 6 different cortical target areas. And that is with a cutoff that is actually relatively conservative about how much axon you need per brain region to be considered a projection. Now, this result obviously rules out the one-to-one model, but despite our best efforts, we could only trace about 50 or so cells, and that was already a large amount of work. But what we found looking at these 50 cells, they all kind of looked unique. Each one had its different features. We were basically in the stamp collection phase. So we didn't have the statistical power to distinguish the two broadcasting models we've drawn up, where you have independent projections or projectional cell types. And so this is where MAPSeq came in, where we really just wanted to bring up the number of cells that were traced so that we gained the statistical power to understand target choice of these broadcasting cells. So we injected our barcoded virus into primary visual cortex. Oops. Well, that's interesting. Into primary visual cortex. Then dissected out 6 higher visual areas right surrounding V1 and identified these by using intrinsic imaging. Sequenced all of these tissue samples and then reconstructed the projection pattern of about 600 or so individual V1 neurons. And what we could do then is count up how often we saw each projection motif, each co-innervation into different target areas. And the result of this you see here, where in the blue bars you see how often we saw each co-innervation or pattern in our data. And we compared this to a null model where we assumed that target choice would be random, basically a coin flip for each innovation into each possible target area. And what we see is that actually there's there's lots of places where we observe co-innovation much more frequently than we would have expected by this random null model, and conversely others where we observe a co-innovation much less frequently than we would have expected by chance. So what that means then is that not only does the average V1 neuron project to more than one target area, and it's therefore a broadcasting cell, but also that these cells broadcast in very specific patterns in essentially projectional cell types where some co-innovation patterns occur more frequently than we would have guessed, and others seem to be selected against. So that's MAPSeq. MAPSeq gives us this, this high throughput for single cell tracing. But if you've been paying attention, you've noticed obviously that because we use Illumina sequencing to read out the barcodes, we lose all spatial resolution beyond what we initially dissected, right? Like we need to grind up the tissue to get the RNA out. So to try to overcome this issue for some applications, Xiaoying Chen, then a postdoc in Tony Zeta's lab, now actually at the Allen Institute as a PI, developed this method called BARSeq, which allows us to in situ sequence barcodes directly in the tissue, so determine the barcode sequence of each mRNA directly in the brain slice. And so what this looks like is something like this, where here you have a dish of neurons, have been virally barcoded, and we perform BARSeq on them. And so the neurons will light up in different colors on the different sequencing cycles based on which base is encoded at that position of the barcode. So if you look at neuron 1 here, it's magenta, or purple, I guess, on your screens. meaning that at the first position of the barcode it has an A. Conversely, neuron 2 is cyan and therefore has a G. And so if you perform the subsequent sequencing cycles, you can follow the change of colors and decode the barcode sequence that is present in each of these neurons. Now what that looks like in the brain is something like this, where here we have an injection into the auditory cortex, and we're just skipping through different sequencing cycles. And so you can imagine that for each cell, if you track the sequence of colors, you can reconstruct the barcode sequence that it's been uniquely labeled with, and then you can go to the projection targets of these neurons, cut them out, sequence them by Illumina sequencing, and then for each of these flashing cells here, you can say, oh, this cell projects to these 3 target regions. You can then take all this data, classify the cells maybe into their projectional cell types, as we've done here, into these 3 cell types you can see on the left, and then look at where these cell types are in, for example, their cortical depth. Now this study is all sequencing the barcodes just at the cell body. Since then, people in Tony's lab have kept working on this and have actually tackled the much harder challenge of sequencing barcodes in the projection targets, where there's just much less RNA to go around. But this seems to be working now. So here you have an injection into auditory cortex, and in white you see all the products of the basic reaction. And so if you zoom in on the auditory thalamus here, what you can do is sequence the barcodes that you find in the projection targets and actually reconstruct each single cell's axonal arbor there by just connecting the dots. And the really cool thing is here that because each neuron in the auditory cortex is labeled by a unique barcode, which you can then recognize in the thalamus, we don't need to do any tracing. We don't need to follow that axon of passage all the way from auditory cortex to thalamus or contralateral cortex. We can just say, oh, we see the same barcode here in thalamus. We know which cell is coming from, and you can just focus on the axonal arbor itself. All right, so that's MapSeq for lots of cells, BASIQ for spatial resolution. How about this multi-omic integration that I promised should be easy, right? Given that the barcodes are mRNAs. Well, one way to do this, and this was recently published by Esther Klingner and from Denis Laboudon's lab in Geneva, is to simply combine MapSeq with single-cell RNA sequencing. So what these guys did is they injected the barcoded virus into developing cortex, labeled a bunch of different somatosensory neurons in cortex with different barcodes, and then dissected out the target areas to sequence them for regular MapSeq. But instead of grinding up the injection site, they took all those neurons and performed single-cell RNA sequencing on them. And what that then gives you is, obviously, you sequence the cell's endogenous transcriptome, as you usually do in single-cell sequencing, but because the barcode mRNA is an mRNA, it effectively looks like an endogenous messenger transcript, it will also get sequenced. And so at the same time as you get the endogenous transcriptome, you also get the barcode sequence that's present in each of these neurons. And so you get your t-SNE plot where you have, based on your gene expression pattern, but then for each dot you also have the projectional information and can add this to these plots. An alternative approach to combining transcriptomics and single-cell projections is BASIQ2, developed by Yuqi Sun and Xiaoying Chen, where they've been leaning on this BASIQ approach, this in situ sequencing approach, to not only sequence the barcode mRNAs in the tissue, but also sequence and detect endogenous marker genes. And so essentially you get your cake and you have to eat it too. Anyway, what you get then is you get the marker gene expression for all these cells in space. For the very same cells, you get which barcodes are expressed in there, and then you know where these cells are projecting to from doing Illumina sequencing, or maybe even BASIQ in the projection targets. All right, so That's the, well, anyway, so all of these methods that I've been talking about so far are essentially still, traditional neuroanatomical methods, right? They're really good at telling you where your cell, where your favorite region is projecting to in the brain. But if you're maybe interested in, more than one target, more than one region, or maybe even network connectivity and the like, what you are forced to do, because you do this only one injection at a time, is inject many different brain regions in many, many different animals, and then do this and trace all these animals, and then collate hundreds of different experiments into one data set. And the Allen Institute has done this beautifully in the Allen Connectivity Atlas, which is a great resource to all of us. But if you want to do this for your own favorite mouse model, that's likely out of the scope of what your lab can finance. And so we thought maybe with this huge diversity of barcodes, right, which I said, should be able to label all the neurons in the mouse brain at the same time, maybe we can do something better. And so this is a method we call BreakSeq that Longwen Wang and I recently published on, which essentially does hundreds of MapSeq experiments inside the same mouse. And so the strategy is as follows. Instead of just injecting a single brain region and barcoding cells there, here we inject as many brain regions as we can. In our proof of principle experiment, what we did is we tried to barcode all cortical neurons in one hemisphere. So we injected 150 sites in the same animal, trying to label as many neurons as possible, as uniformly as possible across this cortical hemisphere. We then let the virus express and used then laser capture microdissection to dissect out all the different cortical regions, both on the ipsilateral injected side and the contralateral hemisphere, as well as in some subcortical regions for ID purposes. And then sequenced all of these in something like 300 what we call cubelets or target regions, and essentially get a matrix of barcode abundance by area. Now the trick is to try to turn this into a connectivity matrix. And what we realize we can do here is actually surprisingly simple to do this, because we've realized that as naive as it sounds, the cell body is really big when compared to axons, right? It has huge volume, axons are super thin, and it turns out that there's a lot of mRNAs, barcode mRNAs in the cell body as compared to the axons. And so when you just look at barcode abundance across the brain for each barcode, you see these spikes in your barcode abundance of like 2 to 3 orders of magnitude more barcode mRNAs than in any of the target areas. And that we can actually show is basically every time the cell body. And so by just going through all the barcodes that we've mapped in this barcode abundance matrix and finding these spikes, we can assign them to the cells to individual areas. What that looks like then is, for one example cubelet, one example area is something like this here, where we have 700 cells or so whose cell body seems to be located in this cubelet 53. I think it's somewhere in somatosensory cortex. And so then you can, what you see here is these 700 cells projections to 100 or so ipsilateral cortical cubelets, the equivalent contralateral cubelets in some subcortical target areas. Now we can summarize all of these single cell projection patterns for this area into a bulk projection, right, by just summing them up. And then you can do this for all the different cubelets we traced in this animal, and you get connectivity matrices like this, where from a single black 6 animal in a 3-week experiment, we mapped out this cortical mesoscale connectome, where you can get the projections from 120 source cubelets in one cortical hemisphere to themselves, to the contralateral hemisphere, and to some subcortical regions. Now this BreakSeq method is fortunately very reproducible. We get nice correlation of connection strength across different repeats of this method in different animals. And when we compare it to our ground truth data, to the Allen Atlas, Allen Connectivity Atlas, we find that our connectivity measurements are about as well correlated to the Allen Atlas as different injections from the Allen Atlas are to itself. Now, the cool thing about this method is obviously not that we can do about as well as we do with other methods, but that it's so fast, right? So like in an individual lab can now in a few-week experiment actually map out large-scale connectivity networks in their favorite model system, in their different disease condition, in whatever you have on your bench, right? And then compare connectivity across these states in what you would call comparative connectomics. To illustrate this possibility, we decided to look at the BTBR mouse. So that's an inbred mouse strain that people use sometimes as a model for autism. But more importantly for us, it has a striking wiring deficit where it doesn't have a corpus callosum. So there's no projections from the one hemisphere of cortex to the other. And so when we take one of these mice and apply BRIC-seq to them, what we see is a surprisingly intact ipsilateral connectivity diagram, but then essentially no projections to the contralateral side. And you can imagine applying these methods now in somewhat less drastic wiring deficits. Now, where do we go from here? Well, there's the obvious directions where we need to keep scaling these things up and try actually to map out brain-wide circuits. So obviously, we'd like to combine BRIC-seq, this brain-wide, super large-scale single-cell tracing with BAR-seq2, so that we can measure both the transcriptional and connectomic cell types of the same cells across, say, all of cortex. The other thing which I'm very excited about is that because BAR-seq is a spatially resolved method, right? You get the 3D location of each of the cells that you've barcoded. You can register this data to anything else that is spatially resolved at that single cell resolution. And the the obvious suspect here would be calcium imaging, so that you can get mouse behavior, functional activity of single cells, and then through registration of the calcium data to in situ sequencing data, also the cell's gene expression and single cell projection patterns. Now, as Linda mentioned, I just started my lab earlier this year at Johns Hopkins, where we're actually leaning very heavily on this idea of comparative connectomics in two different tracks, where in the one we're applying and honing our tools for comparison in different mouse models to understand wiring deficits in disease. And then in a second track, we'll take these now improved tools and our strategies from comparison and apply them to different species to get to use evolution as the lever to understand the organizing principles of brain connectivity. Now, the first target in this first track across disease states, we're looking at wiring deficits in different autism models with the idea that maybe different genetic lesions are causing, or subsets of different genetic lesions are causing convergent wiring deficits that then cause these convergent behavioral phenotypes that we see in various different mouse models of autism. We've recently got funding for this, and so we have a postdoc position available. If anyone's into this, come find me later. The other idea that we're following up on is this cross-species idea of comparative connectomics, where essentially we're asking, how does a brain system gain new functionality? How do you make a new brain region? How do you add a new computation to an already existing system? And what then does this tell us about modularity of the brain building blocks? Is there anything that can simplify this whole mess? We've looked at this in the or we've begun looking at this in the cerebellum, and specifically in the cerebellar nuclei, which are the output structure of the cerebellum and receive all the Purkinje cell output. So without the cerebellar nuclei, the cerebellum is basically disconnected from the rest of the brain. And so the cerebellar nuclei are extremely interesting for us from this evolutionary perspective, because while the cerebellar cortex across species has actually been relatively stable, just expanded and contracted as a laminar structure, the cerebellar nuclei have been extremely plastic. So they occur in different numbers in different species, in different vertebrate lineages, with just 1 per hemisphere in sharks and amphibians, 2 in reptiles and birds, and 3 in mammals. And so if you look at that cladogram, it really looks like these cerebellar nuclei have evolved all from one single ancestral brain structure in the last common ancestor of all clawed vertebrates. And so by doing a whole bunch of single-cell sequencing and spatial transcriptomics work, we could show actually that, at least in amniotes, the cerebellar nuclei are all composed of these repeated copies of just this single cell type module, where we have 3 inhibitory cell classes and 2 excitatory cell classes that occur in these spatially segregated lots next to each other in the cerebellar nuclei and form new output channels to the cerebellum. And this led us to propose that the cerebellar nuclei are actually evolved by a process of repeatedly duplicating this conserved cell type set, this archetypal nucleus, followed by divergence of the projection patterns. Now, how these new modules are integrated into the brain and how they actually support new behaviors is completely unknown because no one's done any of the tracing work. And so we're hoping to apply our barcode connectomics approaches to these different modules now in not just mice, but also zebra finches, reptiles, and amphibians, to really nail down how connectivity changes when a new brain region is born. With that, that brings me to my acknowledgements. Obviously, most of that barcoding work happened in Tony Zeta's lab from Cold Spring Harbor. I've mentioned most of the key players along the way. We're lucky to get funding from lots of places for that. The cerebellum work came from my postdoc in Legion Lowe's lab at Stanford. And as mentioned, we just started at Hopkins, labs growing, and it's coming together reasonably well. So if we need people. If you know anyone, or if you're yourself interested in barcoding, comparative approaches, or this comparative connectomics, come and find me after. And if we have time, I'll be happy to take any questions.
Thank you very much. That was really a fascinating talk. I'm very, very excited, and again, looking for postdoctoral fellows, so please find him afterwards. So our next speaker is Damien Fair. He is a cognitive neuroscientist and professor at the Institute of Child Development and the Department of Pediatrics at the University of Minnesota. He's also the Redleaf Endowed Director of the Masonic Institute of the Developing Brain. He obtained his Master's of Medical Science degree from the Physician Associate Program at Yale University School of Medicine, and he later graduated from the Neuroscience Program at Washington University School of Medicine in St. Louis, followed by a postdoctoral studies at Oregon Health and Science University. In 2012, he was awarded the Presidential Early Career Award for Scientists and Engineers, the PCASE Award, and in 2020, he was named the John D. and Catherine T. MacArthur Fellow. Dr. Fair is deeply committed to public service, community engagement, and STEM education, and his research combines technical advances in functional MRI, advanced mathematics, and engineering, and expertise in psychology and neuroscience in both human and animal models. He is using non-invasive tools as a bridge between species, and he's focusing on mechanisms and principles that underlie the developing brain. So we're very excited to hear from him today. Welcome.
Okay. Well, thank you, and thank you for having me. Glad to be here. So I'll get started. I always like starting off with this slide in my talks with a lot of the folks that are involved with the work that I'll be talking about today, mostly so people can see the amount of effort and level of expertise that's required across lots of different disciplines to make it go. So I have one disclosure. I'm the co-founder and equity holder of Nous Imaging, which licenses the firm motion monitoring software. There'll be one slide on that, so I want to make sure that's everyone's made aware. And I'll go through four specific bins here. One is I'll do a brief history of cognitive neuroscience in the state of our field. I'll talk a little bit about big data, particularly with regard to MRI and big data in many subjects and some lessons learned and some applications. And then I'll do the same thing with big data, few subjects, some lessons learned and applications. And I'll end with a bit of thoughts and conclusions to discuss and talk about our future. So it may actually be hard to believe, but the term cognitive neuroscience is actually about exactly 50 years old, born out of a late night city taxi ride. Now, the idea of the term was meant to kind of take the basic underpinnings of the biology and neuroscience and link it with the intricate dissection of psychological processes. Now, not surprisingly, the field really took off with the emergence of PET imaging and functional MRI. Now, at the time when these technologies first came to be, it wasn't super straightforward what's the optimal way to use them, because they in general had low signal to noise. And it was actually a clinician in Mark Rakel's lab named Eric Riemann who came up with the idea, which seems kind of pedestrian now, of taking individual brains with lots of variability and low signal to noise, putting them onto a common atlas, and then averaging those individual subjects to get a signal strong enough to see some of the underpinnings of the tasks and different types of processes they're trying to. And it was, in the terms of from Gazzaniga, the results of this approach was unambiguous. There's a landmark paper that followed and presented the first integrated approach of design, execution, and interpretation of functional MRI findings. Now, this is actually a picture of as a graduate student, one of my first papers. It's a paper on perinatal stroke, where we were trying to understand and dissect psychological processes related to lexical processing and brain activation. This was kind of the state of the field for many years, up around to about that time. And of course, there was also at the time where there was lots of excitement about how fMRI could be used in clinical settings. I went back and looked at some of one of the papers. This is one of the more fun ones of how fMRI could be used in various ways. So discussions of the future of fMRI have conjured up visions of mind-reading devices used everywhere from front door at the airport terminal to the back room of corporate personnel offices. At least one neuromarketing research firm is already trying to use fMRI to probe what consumers really think about their clients' products. So the idea of what you could use fMRI for at the time was pretty advanced. But while fMRI continues to be extremely valuable in characterizing brain function activity patterns that isolate these various types of cognitive processes, its actual clinical utility is primarily relegated to pre-surgical planning, and even then it's not used widely, largely because it still had this issue of reliability and low signal to noise. Now, around the same time that this was going on, in the background was this idea of, based on a study by Brad Biswell back in 1995, this idea of functional connectivity MRI, where it was a very simple experiment. We had someone go in the scanner in bilateral finger tapping, and you could identify the regions that are related to finger tapping. But then you can have those same subjects go in the scanner, not do anything at all, measure the spontaneous activity, and you can see correlated activity. See correlated activity of those same parts of the brain, while subjects aren't doing any tasks. So this is what, in essence, is resting connectivity. And while it lay dormant for many years, it really took off with this paper in 2005 by one of my classmates and colleagues down the hall, Mike Fox, who's also in the lab of Mark Rakel, which, while this wasn't the first to show these complex systems and how they're designed and organized and how they're negatively correlated, it was definitely the one that probably set the field on fire of this technique that you could use while people weren't actually doing a task to really understand basic fundamental topological and topographical aspects of the brain. This was my actually first foray into this space, and it was me and one of my colleagues, Nico Dozenbach, where we were trying to identify. We had these sets of brain regions in the anterior cingulate and the bilateral insula that were co-activated across many tasks, important for like high-level control processing. We wanted to see if they worked together. So I went down the hall, talked to my buddy Mike Fox about some of the techniques, and we were able to kind of implement some of the technology in the lab to see if these regions were co-activated, and lo and behold, they were. We could put a seed region down here in the anterior insula. You could see the spontaneous correlated activity of these regions that we thought were part of this control system, and it was pretty exciting. But it was so exciting, and of course I was a graduate student, I realized that if I could put a seed region there, I could easily put one over here, I could put one here, I can 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 put one here. We can put these things anywhere, right? And that's exactly what we did. We had, hundreds of these maps littered all over the lab, and there was no computer that could hold them all. We had them all in binders, and but we realized that we had to be able to condense this information, and we came across this paper by Olaf Sporns, computational neuroscientist, on the organization, development, and function of complex networks described with graph theory. I'm not going to go over all what that is, but probably one of the major aspects of that work up until now that has really held strong and has been extremely important is the idea of modules or clusters of nodes that are densely connected to relative to others. And we use these techniques called community detection techniques to be able to identify and classify and characterize these systems. Now, there's many fundamental properties of brain organization and topography and topology that's come out of this work. We now have all these different systems and networks that are now kind of just flow off the tongue as far as like how the brain is organized. And it even, of course, conjured up even more thoughts about how we can we can apply the applied revolution of resting state coming, even rivaled some of those early thoughts of traditional fMRI. But it also shifted some of the questions because of the ease at which the data could acquire. So no longer, instead of doing these repeated measures within subject, samples of traditional task fMRI, which has extremely lot of power, we were now doing these studies of taking complex behaviors and looking at them across lots of different people. It's just fundamentally a little bit different design, types of questions being asked with state characteristics like connectivity and even structural MRI than it is with fMRI. But a potential issue, like I said, is that designs didn't change. But along the way, and over the last 5 or 6 years, as these techniques are expanding our understanding of organization development, the data sets were also increasing in size and or the amount of data collected per subject. So it's probably starting with the Human Connectome project, but and now we have the ABCD study, which I'll talk about in a second. It's 10,000 participants. The UK Biobank, 100,000 participants. It's growing and growing and growing. And then also data, starting with the My Connectome project by Russ Poldrack, who scanned himself twice a day for I think upwards of 14 months, or for 10 months, about 14 hours of data within one person. The Midnight Scan Club, which did the same thing with another 10 people. It's starting to tell us new things. Now, about the same time that we started seeing some signs of failures in reproducibility that started becoming more apparent. And there was lots of papers that have been starting to come out that's highlighting how some of the efforts that we've been seeing are not always reproducible or reliable. So as the field continues to evolve, and data is being collected at a faster rate than ever before, of course, network neuroscience and the view is the brain as multiple interconnected systems that supports complex behavior have also advanced the field in many directions. But the arrival of these large end data sets and this repeated sampling is potentially highlighting, finally, kind of the parameters in the context by which the field can apply some of these lessons that we're learning to the applied and clinical sciences a lot more effectively. And that's, in essence, what I'll be going through now. So big data in many subjects, some lessons learned and applications. So the ABCD study is a study we're nearly halfway through, or about halfway through, of collecting nearly 12,000 adolescents across the United States that's being that is being leveraged for all sorts of things. The data I'll be talking about today is based on a collection called the ABCC, or ABCD Community Collection, that's being maintained by Eric Fezco at the University of Minnesota. It has lots of different data that's been used, that's available for the community, and resources to be able to handle and utilize all these types of data. The project is basically a collaboration across multiple labs, but is really driven by two really fantastic young investigators, Scott Merrick and Brenda Turbo-Clemens, Nico Dozenbach, and myself. But again, like one of these very large efforts where people have to come together for a common goal. Now, the basic question of this particular experiment is it's very simple. So what can we expect? What are the effect sizes of brain behavior correlations if you're doing these kind of state trait characteristics that I was talking about? And does the reliance on typical sizes of data, neuroimaging sample sizes, provide an explanation for why we may be seeing some of these replication failures in the literature? And if so, the question is why? Well, I'm not going to go through all the details of this experiment, but just to highlight that you can do this almost any type of way you want, from on specific connections, specific ROIs, different types of parcellations, with structure, with function, across many different variables. We did several different variables. The gist, in essence, is the same. So what we're looking at here is a relationship or correlation of this functional connectivity data on the right side over here, in the distribution of measures of psychopathology and this functional connectivity. And what you can see, and on the bottom, if I can get this, on the bottom here is just the effect size of the correlation. And what you can see here is that the correlations are relatively small across the sample of ABCD. It's about 4,000 participants. If you look at cognitive ability, it's about the same. If you look at cortical thickness, or it's about the same. The point is that the largest effect sizes are on the order of an R of 0.16 and you can see the median in the top 1%. The point is that the effect sizes are extremely small across these populations. And so the question now is, well, does the reliance on these typical sizes provide an explanation for why we might see some replication failures? Again, I'm not going through the details, but I'll just give you an example. So here what we're doing is we're looking at a distribution of subsampling the data, this ABCD data, into small subsamples. So small subsamples of 25, subsamples of 50, 100, etc. And we're just looking at the distribution of the effects of those subsamples across different sample sizes. Now, if you're subsampling, just as an example, if you take one lab and they're subsampling just a group of 25 subjects, they might find one of those across that distribution. The relationship looks like this, that is quite positive, passes multiple comparisons, it's really highly significant. But another lab might see something completely opposite. And if you look at throughout the literature, you see signs of this in lots of places. Doesn't really matter what you do, the same gist is what you see. And it's because the samples are the effect size are relatively, the true effect size here in the line here is relatively small, and you need to get it right, it looks like you need thousands of participants. Now, here's the distribution of more than 1 million z-values from papers out of Medline. This is the rendition from our folks on Twitter. Okay. But it's not hard to see how you can get an inflated sense of what your effect sizes could potentially be, if you are following the standard practices of what we report and what we accept, into our publications. It's easy to get an inflated sense of what effect sizes should be, considering what I've just told you. So the observed effect size, it seems like for these kind of population study, require consortia-level sample sizes, upwards of 2,000 participants to get the appropriate power. And of course, the question is, well, is this specific to multi-site ABCD study? And just to ease your minds, the answer is no. It's the same for adult and human connectome or UK Biobank data as well. So does the reliance on these typical sample sizes or these sample sizes, cause or provide an explanation for why we have some replication failures? Is probably, and if so, why? The answer is yes. In that this small sample sizes with the sampling variability can produce statistical errors that undermines our ability to reproduce findings. And if you folks are interested in really digging into this, there's a really nice paper by Anthony Dick and Wes Thompson, which really kind of dig into some details on how to think about it. Now, I always put this up because this is really important for most folks. But what these findings do not mean is that small sample studies are not important. You'll see that in a second. That all the foundational work of fMRI of functional connectivity is invalid, and that these effect sizes can't be improved with improved methods and whatnot. But we really need to think about this to make sure that the data are being, that how we're approaching the data are ways that can actually provide important information about brain function, organization. Now, that really reminds me, the state that we're in and what we're kind of seeing reminds me quite a bit of where genetics was when several years ago, or over a decade ago, when we started looking at candidate genes and doing GWAS studies. In fact, I pulled up this review, which was from just over a decade ago, which is not really, not because of the quality, but it really highlights some of the same issues that we're dealing with today, and in that, most of the studies are relatively small, but the power or the amount of subjects required to achieve the appropriate power for different types of effect sizes require much larger studies. And in fact, this is just a little quote from the paper. In this new era of big data and small effects, a recalibration of views about groundbreaking findings is necessary. And I think we're very much at the same spot. Now, one of the things that we've been learning and we've been applying, others, is that after this time, the idea for GWAS studies and doing work that kind of combine a lot of small effects with polygenic risk scores and things like that began to emerge. And it's beginning to emerge in functional imaging now as well, where we can, and structural imaging, where we can combine a lot of these small effects to look at, to provide enough power to even to utilize smaller sample sizes. This is a nice paper out of the lab of Anders Dale and some of his group, kind of highlighting this to be true. And folks have been applying, including us, where you can leverage very large consortium level data, like the ABCD data and the UK Biobank data, to look at relatively smaller samples. And we're starting to see very reliable, very specific effects related to brain scores of executive function and memory, general ability, emotion, how that relates to things like ADHD. You can start to see highly reliable findings by leveraging some of these consortial data to look at smaller samples. Now, there's a few other lessons that we got to learn, and I just always want to highlight this, is that as these data sets get bigger and bigger and bigger, it's extremely important that we make sure the populations that we're studying are really truly representative of our society. I think one of the lessons learned in the genetics world, as they started doing the same things, is not doing that. It can cause some predictive value, issues if the folks are not represented in some of these big data sets. And I think this is a nice paper that really just demonstrates that, and how some of our inequities that exist in our society can actually increase if we don't think about this right up front. So the era of big data has not necessarily been kind to the shifts in study designs and new standard practices and analyses in development of cognitive science. However, after building off some growing pains in genetics, these multivariate polyneural score approaches are allowing for increased power to study brain behavior association in small samples. Doing this work is extremely resource intensive and is unlikely to be conducted by one group alone. But while the work can put us on a more solid footing with regard to fundamental findings in brain organization in population studies like GWAS and what we're calling polyneural, like GWAS and polygenic risk scores, its entry into the direct clinical utility is likely to take a little bit of time. And I think that some of some of that, this type of work, this population work, I think is bearing that clear, at least in the genetic world. But what about the big data fused subjects? Some lessons learned and applications. So I think let's start with traditional structural MRI. Everybody knows MRI, structural MRI. Folks typically understand how it's used, or it's definitely used in the hospital. It's used for all different types of ways in the clinic. There's no question that it has this the clinical utility is vast and widespread. Let's just start with, well, how did that happen? And why hasn't techniques like functional MRI naturally followed that trajectory? So one is that, well, structural MRI in individuals can be used effectively and reliably to detect variations from person to person. It's extremely high contrast, what we call contrast to noise. It definitely can assist in adjusting treatment or the options. In my previous life, I did stroke neurology, cerebrovascular disease, and so in the ER we use CT and MRI to make big shifts and adjustments in treatment. The effort is generally low on collection and on the patient, and in many cases there can be a reliable response to some type of intervention. Okay. All right. So, like I said earlier, almost most of the work that we've been used to examine, try to understand some fundamental principles in functional organization using these techniques like resting connectivity MRI has been based on the average. Okay. It's based on taking a bunch of subjects, generating their matrices, averaging them all together, so we have enough signal to actually look at the specific topological phenomena. Okay? But of course, nobody's average, right? Everybody's a little bit different. Now, I wasn't exactly sure how this was going to play in this particular academic crowd, but for those of you who don't know, LeBron James is a very famous basketball player. Now he plays in Los Angeles. And of course, you must all know who J.J. Barea is, not because you're in Puerto Rico. All right, so like I said earlier, Russ Poldrack kind of started this off with this MyConnectome project, scanning himself for upwards of 14 hours over the course of 10 months. And using that data, was able to really highlight that a lot of the same fundamental structural or functional organization of an individual brain looks much like you would see in an average brain, but there's lots of different, very specific, precise differences in his brain relative to the average. The nice thing about these types of data sets, where they're so large that you can show very specifically that individual's fingerprint is highly reliable and completely replicable from study to study, okay, using this kind of functional precision mapping. The Midnight Scan Club took this one step further and did, in essence, the same thing with about 5 hours of data per participant in 10 different subjects, and reaffirmed some of those initial findings, but showed how the variability is from person to person to person to person. Very specific deviations and organization distinctness for person to person is all there, even if the general structure is there as well. Now, this is a case of a child with a very large bilateral perinatal stroke. Didn't actually know he had a perinatal stroke until he came into the neurology department after he was a teenager and being a pitcher on his baseball team. Now, what I'm just trying to highlight in here is not the entire study, but specifically in this purple here is this outline of the connectivity boundary. It's just highlighting that these types of studies where you have hours and hours and hours on a given individual can precisely map exactly how his brain, this child's brain, is doing his motor functions relative to the average, which is over here. The blue is our child, where his motor representation is, and this is what you typically see in a group of controls. So the point here is that with these new techniques and these new precision mapping, you can effectively, reliably tell variations from person to person within a subject. Okay, well, what about responses to intervention? There are some evidence that this is true too. Now, this is another example of a study where, which is building off some of the early sensory deprivation work by Stryker and colleagues to induce neuroplasticity within a specific person. The way the experiment goes is you cast the dominant arm of a given individual for 2 weeks, and then you do serial scanning daily, about 42 to 62 separate scans over time, before and after and during the casting. And what you can see, this is just the seed region over here, which you can see a couple of things. This is this is just one subject, but this is the pattern of the connectivity in that individual. On here, you can see in the contralateral, you start to see some decoupling of the connectivity, and this happens over the first 48 hours. But also interesting, you see high connectivity, within the region that starts to increase, which comes back to normal after you take the cast off. Now, this is highly replicable from person to person to person. The timing's about exactly the same, but this increased connectivity is also highly interesting because it seems to be driven by these very specific bursts of activity that occur kind of spontaneously, only during the cast period, and only during the cast period, and only during the contralateral cortex. Now, what's nice about this is it reminds me very much of some of the bursts of activity that you see in the development of some of the historical, ocular dominance columns, the work by Huber and Wiesel during the same types of sensory deprivation. And so the advancement of the imaging, which is paralleled in some of, I'm not going to show it today, but it's being paralleled in some of the animal models from non-human primates to rodent models, is starting to allow us to be able to tackle some of these issues, or these ideas, these hypotheses. So there's some evidence that the technique now can respond to interventions. Can it assist in adjusting treatments? Now, there's not a whole lot of data on this, but there is some things that are coming out. So transcranial magnetic stimulation, which is FDA approved for the treatment of depression, for treatment of depression, can show that you can show that the specific target of trying to hit the DLPFC, the dorsolateral prefrontal cortex, that connects with the area of the subgenual cortex, that the precise location of that targeting with TMS improves if you hit the precise target for that specific individual. This is just replicated just in a couple of weeks ago with a paper by Siddiqui et al. So it's beginning to show that for some types of targeting, if you have the precise location, then you can improve some of the outcomes. There's also some data showing in the subcortical realm, some of these locations in the subcortical regions that are highly integrative across different complex systems in the cortical range are also potential prime areas for targets. And as we begin to expand on the use of deep brain stimulation, MRI-guided ultrasound, being able to utilize some of these precision mappings to help guide some of those new ideas and those new therapies could be potentially beneficial. And I think this paper by Skangos et al. out of UCSF is really, it's really kind of a nice example where you put multiple intracranial electrodes along the limbic system in a patient who's depressed, and you can very precisely, reliably, quickly be able to start changing mood and emotional states by using a precise location for that of stimulation for that individual. So again, like combining these techniques, you can see it kind of coming. All right. Now, the last piece here is the effort on collection. I've already told you can kind of get a sense that the effort to be able to collect this type of precise data is extremely, extremely high, right? They're collecting hours and hours of data within given individuals to be able to get this precise map. And indeed, if you want to get to approximately 90% of the max or so, you need somewhere between 40 minutes and hours of data within a given person to be able to get their precise map, to get enough signal to noise so you can see those types of changes in an individual. So that's kind of high. Now, of course, there's lots of work going on right now to try to reduce that time. So the work using multi-echo MRI is a nice paper by Lynch et al., which is highlighting that there's ways that you can acquire the data and process the data that can potentially speed up that time. What we're doing is trying to improve the workflows and providing feedback to the patients to help reduce the time and make it a lot easier to collect these types of data and reduce the burden. But what if maybe you don't have an MRI, right? Or what if you can't, no matter what, you can't get the patient to stay in. I do studies with ADHD. You can't get the patient to stay in there for 2 minutes, right? What do you do then? So one of the things that we've been working on is the idea of generating probabilistic functional atlases to hopefully handle handle that particular problem. So again, like the gold standard one of the gold standards for generating these systems and networks in the brain is to use this community detection technique. Well, we've been kind of playing with this idea of what we're calling template matching, where it's more supervised way, where if you have some prior of understanding what the network should look like with an individual, can you use that prior to be able to identify the individual network organization for a given person? So in essence, you take a given voxel, or what we call gray ordinates, and you compare it to a specific template. In this case, it's a winner take all. And you see if you can generate some of these individualized networks. And there's two goals that I'm going to talk about right now. One is that we want to validate this technique, relative to what we say is the gold standard, and then determine if the amount of data that's required is actually less to get an individualized, personal, precision systems. So to do this, you can utilize some of these data. This is that midnight scan clump data where you have two halves of data, approximately 2 hours each, and one half is you can use to help validate any type of test, and then you can use mutual information based on to see how well these things relate. And of course, that can be looking at one technique versus the other, like community detection versus community detection, or you can do community detection versus template matching, or template matching versus template matching, etc. So here I'm mostly showing you the relationship between this new technique and kind of the gold standard. And this is just one participant, and just highlighting some of these specific variants that exist in a given individual, and how they seem to pop even with this technique that potentially requires less data, and is in a supervised fashion. Here is actually looking at all the participants in the Midnight Scan Club. These are the distributions of how well a given person's system maps onto themselves versus somebody else, with the normalized mutual information, highlighting that indeed this technique, at least with this amount of data, is able to recapitulate some of the same systems. Well, how much time does it take? So now you can do the exact same thing, except for now you're just comparing the amount of time per person. And the gist is that the blue here is is the distribution of the relationship between one half and the other if you're using all the data. And here's as a function of time. And what you can see is that even somewhere between 10 to 20 minutes, you're not reaching the max, but you're at least within the distribution, suggesting that even though it's not perfect, you're starting to get pretty close. Now, what I'm showing here, this is a bunch of subjects with ABCD. In ABCD, it's a bunch of kids. We only collected 20 minutes of data total. Here, we're looking at about, I think, 10 minutes of good data and identifying their individual networks. And I'm putting a box with the DLPFC here, just showing you like how variable from person to person to person to person these systems actually are, and why you might imagine this precision targeting may be pretty important. Now, you can take this one step further now, and instead of looking at everybody, subject to subject, you can now ask the question, well, what's the probability that at this particular location in the brain, it's this system across individuals, okay? A probabilistic map, what we're calling a probabilistic functional atlas, okay? Here are just a couple examples of different systems in the brain of what these functional probabilistic atlases kind of look like. There's some really good news. So if you try to replicate this across different samples, that the locations, the probabilities across people is highly reliable. Even though relative to the gold standard, we don't have enough data to do it well, but we can see that the results are strongly correlated to the gold standard as well. And you can identify these probabilistic locations in subcortical structures as well. Typically with MRI, because the signal to noise can be even worse as you get towards the middle of the brain, being able to do this is actually quite important. Now, here's just a quick teaser of what this particular target, in the case of TMS and the DLPFC, what it looks like, what that connectivity looks like across individuals in two different data sets, the ABCD and the Midnight Scan Club data, and how that relates to that specific circuit trying to hit the subgenual cortex that you can see reliably across here. Now, if you move outside of that probabilistic zone, and you even under a centimeter, you can see how unreliable it gets from person to person. So we think that using these kind of probabilistic atlases, while not perfect, in the case of really high burden to get these specific targets, that this may be of use. The last part here is thinking not only of these networks as specific entities, but that they may have some overlap. We talked a bit earlier about these idea of these integration zones. And just to make a long story short, what we have using that same type of technique as template matching, you can utilize them to identify networks kind of independently of each other, which allows for specific overlap. Now here's just a picture of these template networks in an individual. I'm just going to highlight one of them for the fMRI consumers. This is the ventral attention network, which typically, I'm kind of highlighting the typical regions of the ventral attention network, but in using this technique, you can identify regions of the brain related to this system that are non-traditional. Now, if you overlap all those different networks and you see where these different networks overlap, you can identify these specific integration zones. Like I mentioned earlier, you have some locations in the brain which have upwards 8 to 9 systems that actually come together in one location. And again, these particular locations, just like I mentioned earlier, are prime targets for various types of neuromodulation. And just like with the probabilistic atlases, you can do something similar with these integration zones by looking across the average, and you can see some of the fidelity and some of the identify specific subcortical nuclei and whatnot. It's pretty fascinating. All right. So while we don't have everything perfect, that you can start to see the wheels turning, that we finally have a place where the data and the fidelity and some of the methods and approaches can start reaching some of the potential that traditional structural MRI has in the past. Now, we've been putting together to kind of push the field in this direction. We've been putting together a lot of resources for them. We've now developed a website and databasing system for folks to be able to get these individual systems, be able to use the probabilistic maps and generate regions of interest, supply their own if they want, and all in order to make the ease of use and the ability to kind of leverage some of these individual maps in their own work a lot easier. All right, so I'm gonna end here with just a couple of things. Typically, in the past, my few talks, what I've been doing is a full-on SWOT analysis. You know, strengths, weaknesses, opportunities, and threats of our field. You know, how we're going about our work, and how to move forward in the future. I'm gonna keep it really tight and short today because of some of the time constraints. Just a few things, but if folks are interested, we have a new review out that describes a lot of what I'm talking about today and more. So there's some general conclusions that I think is important. One is that the field continues to evolve as the data are collected in its broader populations at a faster rate than ever before. Leveraging personalized precision mapping approaches for today's questions is likely going to be a critical step for progress in the cognitive neurosciences and for its clinical utility. We now understand the parameters and the context to which to provide these reliable, replicable findings using functional imaging at both population level, you know, the big data mini subjects, and at the individual level, the big data, you know, few subjects. And that maximizing this potential for utilizing this technology to guide the applied and clinical applications will depend heavily on the field recognizing the strengths and weaknesses of the techniques and challenging some of our institutions that are not necessarily optimally equipped to push the science in the right direction and to provide the correct incentives so we can do the science right. So I'm going to end there. I want to thank, of course, a lot of the folks in my lab and all the funding that went into the work that you saw today. Thank you.
Thank you for that wonderful talk. It was really eye-opening. Our final speaker today is David Boas. He is professor of biomedical engineering and the director of Neurophotonics Center at Boston University. He received his bachelor's in physics at Rensselaer Polytech Institute and his PhD in physics at the University of Pennsylvania. He's the founding president of the Society for Functional Near-Infrared Spectroscopy and the founding editor-in-chief of the Journal of Neurophotonics, published by SPIE. He was awarded the Britton Chance Award in Biomedical Optics in 2018 for his development of several novel, high-impact biomedical optical technologies in the neurosciences. He was elected a fellow of AIMBE, SPIE, and OSA, which are 3 international societies for optics, photonics, and biomedical engineering in 2017. His research focuses on neurovascular coupling, cerebral oxygen delivery and consumption, functional near-red infraspectroscopy, and physiological modeling invasively and non-invasively at both microscopic and macroscopic scales in animals and humans. So studying functional brain recovery in survivors in stroke was one area in which he's been highly successful in applying these translational technologies. So we're really pleased to have him speak with us today. Great. So thank you very much.
Great. Thank you very much for the introduction. It's a great pleasure for me to be here today to tell you about functional near-infrared spectroscopy and how it may be useful to you. I am a co-founder of a company that is working on a different technology in a different population, so nothing related to this presentation. So I've been working in the field of functional near-infrared spectroscopy for well over 20 years, developing the technology, signal processing methods, and helping others adopt this technology. In the recent years, just the miniaturization of electronics and more powerful and smaller computational methods have really driven this ability to do wearable functional neuroimaging, and we were very recently lucky to get a nice Brain Initiative grant to really push this technology further. So I want to acknowledge the NIH for that funding, Alexander von Lumen and Miriam Uchel, who were instrumental in pulling that proposal together, Bernard Zimmerman, who's doing most of the technology development, hardware development I'll tell you about, along with Antonio, Joe, and Anderson, as well as Priya, Deja, Yuan, Jesse Anderson, who have been really working on the testing in human subjects, as well as a number of BU faculty who are helping us apply it to many different interesting applications. But first, let me give you some background. So as we were just hearing from the last speaker, fMRI has really had profound impact on our understanding of how the brain works. But it's limited in how we can measure those subjects. So subjects have to lay down inside the bore of a magnet. So that really greatly limits the types of activities that they can engage in, and in particular, natural behaviors in ecologically valid settings simply cannot be studied with fMRI. This also limits the types of subjects we can measure and the patient population. So you can't measure everybody in an fMRI machine. No doubt over the next couple of decades, fMRI is still going to continue to have just profound impact on our understanding of how the human brain works. But I believe that wearable functional neuroimaging in the next couple of decades is going to really advance our understanding of what makes us human, which is the general social interactions that we have. And so functional near-infrared spectroscopy, in particular combined with EEG, will be quite powerful for this. Both are wearable, portable. FNIRS measures the hemodynamic signal, just like fMRI, looking at integrated brain activity. EEG is able to look at the electrical potentials, more direct measure of the neural activity. Both permit these measurements in unconstrained environments. So commercial products are appearing that combine FNIRS and EEG. It's still very early stage. More optimization is needed. This is aim one of our proposal. Integrating all of this, the analysis methods, into unconstrained environments, real-world type measurements. We're going to need to integrate with eye tracking to help capture the stimuli that are being presented with the subjects. And then, of course, moving from well-controlled lab settings to everyday world type settings, how do we do experiments in those settings? So that's another aspect of what we're going to be working on. But first, let me give you a brief history of FNIRS before I tell you about the specific advances that are happening in the field. So near-infrared spectroscopy first appeared in 1977. Franz Jobst showed that you could shine near-infrared light through the adult human scalp and skull and interrogate hemoglobin concentrations in the brain. So this was quickly adopted during the 80s, in particular applied to infants. Cerebral oximeters developed, and throughout the 90s, and into today, cerebral oximeters are well commercialized, and they're proliferating for monitoring cerebral oxygenation during anesthesia and in patients in the intensive care unit. It's based on the principle of the absorption of near-infrared light by hemoglobin. That use near-infrared light because, well, the visible light is strongly absorbed by hemoglobin, but in the near-infrared, it's much more weakly absorbed by hemoglobin, and so light is able to propagate through the scalp and skull. Using 2 or more wavelengths of light, it's possible to probe the differential concentrations of oxygenated and deoxygenated hemoglobin. So in 1993, just a year after the first fMRI studies appeared, 4 different groups showed that you can measure hemoglobin brain activity responses using near-infrared spectroscopy. And so this is an example from one of those first papers showing during a cognitive task that you could see an increase in oxygenated hemoglobin, a decrease in deoxygenated hemoglobin. Here's a second group that had multiple regions that were measured. They could show that spatially localized responses to, again, an arithmetic task in this case. So basically what's happening, first, FNIRS. So we are putting light into the brain. That light propagates through the scalp and skull. It reaches into the brain. You have a detector that's receiving the light that's diffusing through the head, and the product of those 2 probability distributions gives us a sensitivity profile. So if you have a source and a detector then placed a couple centimeters away, 3 centimeters is typical in an adult human, you detect light that is sensitive to absorption changes in the scalp, skull, but also the brain. Right, so we want to disentangle the hemodynamic fluctuations happening in the scalp from those happening in the brain. And so presenting stimuli to the subject, would preferentially cause manipulation of the hemoglobin concentrations in the brain. So that diffuse light we're measuring penetrate deep into the brain, so we're principally just sensitive to the superficial cortex. But a great deal of brain activity does happen in those brain regions. So how does this relate to neural activity? Just very briefly, neural activity results in arterial dilation. Basically, there's a release of neurotransmitters that are vasoactive. They cause arterial dilation. Blood volume increases as a result. Blood flow increases, delivering more oxygenated hemoglobin to the downstream capillaries and veins. At the same time, you have an increase in oxygen metabolism to produce more ATP, to repolarize the membranes. That decreases the oxygenation of the hemoglobin. The net result is that we always see an increase in blood flow. You see an increase in blood volume, and also an increase in oxygenated hemoglobin. So this is a very typical sort of hemodynamic response function you'd see, where red is oxyhemoglobin, blue is deoxyhemoglobin. People have compared this with fMRI, and this is now just normalizing all of those plots. You can see that the fMRI is measuring deoxyhemoglobin, as we expect from the biophysical models of deoxyhemoglobin, of fMRI bold signal. But it's interesting in this, you can notice the oxyhemoglobin is a larger response. So for statistical significance, we like to look at the oxyhemoglobin signal, and that's just a very simple physiological reason for that, in that the blood flow increase and the blood volume increase are both adding oxyhemoglobin, but for deoxyhemoglobin, they're subtractive. They cancel each other out. Okay, during the first 10 years, 1993 to 2003, there were just 150 papers published. It was easy to follow the field then, read every single paper. And it was quite interesting to look back at this and see that basically papers were published in kind of all aspects of functional neuroimaging. During the next 10 years, the field continued to grow. That was harder to read all of the papers in the field. There was continued technology development and more applications. There's a nice special issue in Neuroimaging 2014 that still serves as a very nice textbook, essentially, of the state of the field. And it's still useful to read today. So I just want to cover briefly some of the areas that FNIRS is being applied, and then I'll come back to the technology we're working on. So just briefly, so one exciting area that's developing is using FNIRS to look at ADHD, which an objective evaluation often involves a neuropsychological exam, but you can also do some neurophysiological testing. So the neuropsychological exams often yield mixed results and are not direct measures of the mechanisms of ADHD, but looking at the effects of the underlying causes. So neurophysiological testing, perhaps will provide a more objective measure of the mechanisms leading to ADHD. And so EEG, evoked potentials during a go-no-go task, correlates with inhibitory function in the prefrontal cortex, and that has been shown to have a decreased amplitude and longer latency in ADHD. Functional near-infrared spectroscopy at the same time has shown a decreased hemoglobin response, oxyhemoglobin response, amplitude during a go-no-go task in the prefrontal cortex, suggesting that there is decreased prefrontal cortex activity, perhaps indicating less inhibitory control. So there was a recent study I'll just tell you briefly about that was combining FNIRS and EEG and just exhibiting this effect. And during this go-no-go task, a subject has to press a button when a target appears on the screen and not press a button when other targets other stimuli appear on the screen. And so with EEG, between a typical developing control and an ADHD subject, you can see very subtle differences in the latency of the evoked potential and small differences in the relative amplitudes. When you look at the functional near-infrared spectroscopy response, you can see actually a profound difference. So in this images shown here, you can see there's an increase in oxyhemoglobin in the right and the left prefrontal cortex. In typically developing controls in ADHD, there's actually a decrease in oxyhemoglobin, suggesting that there's actually less activity and so less inhibitory control. And so you look at the significance of that, it appears to be much more significant than the effects you see with ERPs. And so then others have taken this and then looked at the effects of drugs, and so trying to renormalize that prefrontal cortex activity. So looking at controls, you see the same no-go task. You can see that activation in the prefrontal cortex. In ADHD, after washout of the drugs, you see there's no activity, but then the drug is given, and just an hour later, 2 hours later, you can see restoration of that prefrontal cortex activity. So quite interesting that you can follow these pharmacological manipulations and see restoration of that activity in the prefrontal cortex. So, this neurophysical measure of the pharmaceutical normalization of inhibitory control, could have important use in personalizing the medication for the subjects. And we can expect that technological advances that I'll describe to you later on will really help us move this from the group level analysis down to the individual subject analysis. So another example is using FNIRS to assess depression and schizophrenia. Very similarly to ADHD, the prefrontal cortex is implicated in a wide variety of psychiatric disorders. And so in depression and schizophrenia, this model of emotion regulation and the role of the prefrontal cortex in regulating emotion is important. And when you have dysfunction in the prefrontal cortex, so you don't have the activity providing the inhibitory control, perhaps the limbic system can be hyperactive and lead to some clinical depression and anxiety. And so having wearable tools to measure this in patients could have broad utility. So this was a study published this year that did a very nice meta-analysis looking at FNIRS in conjunction with a verbal fluency task. They looked at 121 papers, and they brought together the results, and essentially 90% of those papers showed hypoactivity of the prefrontal cortex associated with major depression and schizophrenia. And they showed in this meta-analysis that the hypoactivation spatial distribution was able to distinguish schizophrenia and major depressive disorder. So it was a very clever study in which, oftentimes FNIRS, because a lot of people early on with the technology didn't have many sources and detectors that are measuring one region or another. So this meta-analysis very cleverly brought all the data together and assigned papers based on which regions were being measured. And so what they found was, very strong correlation, a positive for hypoactivation across all prefrontal cortex regions, across many of the papers. And then more importantly, when they looked at the ventral versus dorsal responses in the prefrontal cortex, they were able to spatially discriminate signatures related to depression versus schizophrenia. So I firmly believe that with further technology developments, as we get better spatial resolution, with overlapping measurements that I'll be telling you about, it will really improve individual diagnoses, and it'll be interesting to see if this has utility in looking at pharmacological normalization of these prefrontal cortex signals in these patients. So one final example before I go into new technology, this is a project that I was involved in with David Borsick and Miriam Uchel, looking at pain assessment in the operating room. So David Borsick is a pain specialist who tells me that up to 30% of the patients undergoing a surgical procedure will suffer from post-operative chronic pain. This is related to not having enough analgesia during the surgery, and that anesthesiologists need an objective measure of the analgesic load. People are focused on measuring anesthetic load, but not analgesic load. And so he's been studying brain responses using fMRI for a long time, looking at the brain response to innocuous versus noxious painful stimuli. Can't bring an fMRI machine into the operating room, but you can bring an FNIRS machine. And so we've done a number of studies over the years looking at somatosensory cortex versus prefrontal cortex responses to innocuous and noxious stimuli. And so we can see sensory responses differential to the pain level. Also prefrontal cortex, we can see just a profound decrease in activity in response to a painful stimulus. And then we started to look at the effect of an opioid on that painful response, as would be used in an operating room. When you use a placebo group and you provide a placebo, there's no change in the frontal lobe activity. But when we use morphine, you can see over the course of an hour that frontal activity response to the painful stimulus disappears, and then 30 minutes later it begins to return as the opioid washes out. So I think that will have utility, again, eventually in the operating room as this technology advances further along that translational pathway. One thing I want to point out is just one signal processing trick that was really needed to make that work was to look at the systemic physiological response that happens during some types of stimuli, particularly a painful stimulus, that can produce just a systemic blood pressure response. And near-infrared spectroscopy is measuring what's happening in the scalp as well as the brain, and in fact is much more sensitive to the scalp than the brain. And so when we looked at that, just the typical way of analyzing the results, we kind of see this weird pattern that doesn't look like brain activity. And we see it at short separations as well as long separations. So basically, it kind of suggests we're not measuring the brain. In fact, we're measuring a scalp response to the painful stimulus. And so it's important to do what we call a short separation regression, where we take a measurement at a short separation, we regress it out of the long separation and estimate the brain activity simultaneously. And when we do that, this is looking at a response in the somatosensory cortex, we get a very clear localized brain response now that we've removed that contaminating scalp response to the stimulus. And so this is one of those technological advances that will propagate into the field, into more studies that will make FNIRS measurements of brain activity more robust, at the individual level. So during the last 5 years, the technology has continued to grow, more and more publications. Very exciting to see this. And if you are interested, our Society for Functional Near-Infrared Spectroscopy is going quite well, and we'll have the next conference in Boston next October, and so hope to see some of you there. But the last 5 years, I'd say really there's been a marked advancement in the development of wearable technology for FNIRS and multimodal combinations, particularly with EEG. And so that brings us to the Brain Initiative Award that we are now working on, where we are trying to really advance the integration of FNIRS with EEG and promote its use in everyday world settings. So we are removing the traditional constraints of fMRI, and so this will greatly enhance the repertoire of controlled studies that we can do in the lab, but also allow us to do studies outside of the lab in the everyday world. And importantly, we want to study, subjects that are interacting, perceiving the world around them while perhaps they're walking. And individually, each of these is hard to do with fMRI, and certainly any combination of them is very hard to do. And all of these are impaired in patients with neurological disorders, pointing more towards the need to have neuroimaging technologies that allow us to look at these neurophysical measures of brain activity. So I'm working with Alice Cronin-Gollum and Terry Ellis at Boston University, who are studying abnormal gait and walking in Parkinson's disease patients. And they're starting to use this technology. Working with Swathi Kiran, who's looking at aphasia patients and helping stroke survivors learn to communicate again. And David Summers is an expert in perception and attention and working memory. And he's starting to use this technology to complement his fMRI studies of attention and working memory. So before telling you some of their initial results as they begin to adopt the technology. Just a bit of my own evolution with the hardware. I've been working on this, as I said, over 20 years. Developed our first instruments, was shown here. Continued to evolve this. Worked with a company that, a contract engineering company, built us this beautiful system. I think this first system we must have published 40 papers with. And then this system we probably published maybe 100 papers with. This system was distributed to many places around the world, and very excited to see that help the field grow to where it is today. So back then in 2004, the hardware was way more advanced than the experimental methods and the analysis methods. So we focused on that for the last, for the next 15 years. But in parallel with that, just the miniaturization of electronics and the computational power and miniaturization of computers just was just mind-blowing. And as a result, it became possible to put everything you saw in that large instrument, into a much smaller wearable as depicted here. So we can now put the sources and detectors directly onto the scalp with all of the electronics necessary held on a head cap. And so many groups are advancing these methods now, and we're seeing more companies develop wearable products as well. But so it's very early in these stages, and there's much more optimization to do. So we got back into the hardware building business ourselves, and first got a nice small NIH R24 grant to develop our first wearable FNIR system and distribute. We're working on distributing some of these systems to a number of collaborators, and then that made us realize we could make these electronics even smaller. So here's a detector, there's a source, much smaller than a penny. So our goal now is to really populate the head with over 100 detectors and sources. We like to use a hexagonal array. This gives us, importantly, overlapping measurements, and I'm not going to go into the details, but essentially this will help us improve our spatial resolution by more than a factor of 2. So we can expect to achieve spatial resolutions of 10 to 15 millimeters on the cortex versus what has traditionally been the source detector separation is the spatial resolution is about 30 millimeters. So this should greatly aid in differentiating fine scale brain activity patterns. We're using a hexagonal pattern. Others are using a grid pattern. And so there's interesting differences there. We'll see at the end of the day which is the most optimal approach. A grid pattern gives you more overlapping measurements, but it needs more multiplexing. And so it's gonna, the hexagonal pattern will actually have some benefit on weight and battery life and duty cycle that may be more important for everyday world type settings. Okay, so now a really important key advance that's going to happen the next couple years is making these FNIR systems with high density, large numbers of sources and detectors that will greatly improve the spatial resolution. And so we've been working on that as well as others. My former postdoc, Alexander von Lumen, last year went to work with NIRX in Berlin, and they have a very nice wearable FNIR system, not high density. So he shipped me one, and so the first thing I did with it a year ago was actually try to do a high density measurement, and did that. We built this cap, we 3D print this cap, and I put it on my head. This was last Christmas time. And did a finger tapping task and was able to get a very nice localized measurement of the oxyhemoglobin in my motor cortex. What we want to do now, though, is go from, this is their wearable system. This has 16 sources, detectors, 7 sources. It's like one of their systems. It's this big here. So you can imagine, you can wear 4 of these. It becomes quite bulky. And if you, that still wouldn't give me enough sources and detectors to cover my whole head, which is what I really want to do. So we want to populate the whole head with sources and detectors. We've prototyped this. This is what it would look like. But now we need to build the control unit to drive all of those sources and detectors. And so the hardware Bernard Zimmerman is working very hard on that. We expect to have by summertime the electronics to control all of those sources and detectors, so we can measure, whole head brain activity patterns. So what are we going to do with this? So coming back to the walking perception and interaction and the work my colleagues are doing. So we want to look at abnormal gait in Parkinson's disease, language impairment following stroke, and attentional and working memory errors as some neural signature of mental state. So in terms of walking, actually first let me talk about Lou Awad and Regina Slutsky's work. They're looking at post-stroke gait deficits due to impaired propulsion. And so we have these biomechanical measures that can very easily measure biomechanical differences in gait following stroke. And so, these stroke survivors who've had a stroke that affect their ability to walk, their ability to, the automaticity of their walking is greatly reduced. So this can be well measured. Lou Awad and others, they're developing exoskeletons to rehabilitate these stroke survivors that help them get better walking ability and improve their automaticity. But actually, many of these subjects, patients, they literally can't walk and chew gum at the same time. They can't think about their shopping list, for instance. They have to think about all of their muscle movement just to flex their foot the proper way to take each step. And so another goal of these exoskeletons is just to reduce the cognitive load of walking so that subjects can be thinking about other activities at the same time. And so people have used FNIRS actually to look at cognitive load during walking. So if you have a healthy subject that's walking, you actually see reduced prefrontal cortex activity. And when they're walking while thinking, in this case probably doing a mental subtraction task, you see increased cognitive load. In a stroke patient, you can see, though, that just walking has an increased cognitive load. And so they have very little reserve for walking and thinking. And so the idea is this exoskeleton could help bring just this walking back down to what we see in healthy adults. And so to do that, Lou and Regina, they want to move from treadmill-type measurements to walking in everyday world settings. And so they're taking our initial devices with the exoskeleton, and they're beginning to do studies of subjects walking initially down hallways, but then ultimately in more everyday world settings. So they've just started doing this. They are getting measurements of brain activity. This is from one subject. This is a group level analysis of 3 subjects. And this is beginning to go very well. And they're moving to patients, and so we'll start to see some patient data very soon. This is very much related to using FNIRS in Parkinson's disease. This is work that Terry Ellis and Alice Kroner-Gollum are leading with their trainees, Janie Gurnis and Rini Kaplan. And a very similar idea in that Parkinson's disease affects automaticity of movement, particularly with rhythmic tasks like walking. So to compensate, the patients must allocate increased attentional resources to help them walk normally. And so FNIRS can be a very useful measure for looking at these attentional resources that are being devoted to help these patients walk. And so it's very common to do a dual task stimulus on these subjects, have them just walk or walk while counting backwards, and you can look at their biomechanical measures, but you can also measure prefrontal cortex changes in oxyhemoglobin. So my colleagues, though, want to move from treadmill tasks to doing this in everyday world settings so they can look at more natural interactions of the subject with the environment. And then more importantly, they want to develop therapeutic approaches, interventions that can help their subjects gain better automaticity and use the FNIRS as a measure of cognitive load to see if their interventions are effective. So it'll be exciting to see how that develops. As for interaction and perception, a really exciting area that depends on these tasks is looking at autism, which we all know is a complex neurodevelopmental condition with one core impairment being social communication and interaction. So language is one area that is a strong predictor of later social development, and as such, measuring language in these individuals is an interesting and useful target. But it's well known also that individuals with autism demonstrate better skills in more naturalistic contexts. So studying them in an fMRI machine may not be the best thing. You really want to do it in a much more natural setting. And so my colleagues Helen Teger-Fussberg, Lindsay Butler, and Meredith Bekonis are beginning to look at language tasks and just strings of sounds, phonological tasks versus semantic tasks, looking at the FNIRS responses that they're measuring to these different tasks. They're doing this just in a control group now, getting good results, but then they're going to be moving to patients, and more data will be coming soon. So with that, I hope I've given you an idea of the just the growth of the field of functional near-infrared spectroscopy, that just electronics have been driving just a real revolution in the miniaturization of functional neuroimaging methods, in particular FNIRS and FNIRS combined with EEG, and that this is really going to have a profound impact on our ability to study the socially interacting brain while subjects are walking around, real-world environments. And now it's really nice for me to see the hardware for measuring neuroactivation, again, way ahead of the experiment. So the next couple of years, just these advances in hardware are going to drive a real need to develop the experimental methods to help us analyze that data. So, a lot more exciting work to come. So thank you for your attention.
Thank you very much. That was a wonderful talk. Could our speakers please join us on the podium, and we'll have time for a few Q&A. Thank you so much. Thank you. That was wonderful. So I'd like to start with a question from our virtual attendees, so others of you can come up also if we run out of time. So this is from Supash Pandey. He said, very fascinating findings presented by Dr. Zhang. Congratulations in your single-cell approach of transcriptomic and epigenomic profiling. Were data merged to define the molecular taxonomy? If yes, then how much there was overlap between the transcriptome and the ATAC-seq peaks to understand cell-to-cell interactions in given brain regions?
Is this on?
Yes.
Can you hear me well? Yeah, okay. Yeah, that's a great question. So far, we are able to integrate the single-cell transcriptomics and RNA-seq and ATAC-seq data well together. The coverage of the ATAC-seq data is usually sparser. Just by if clustering by itself, it's actually pretty challenging in identifying all the cell types. But if the ATAC-seq data can be mapped into the RNA-seq data, the resolution becomes a lot better, and we are able to identify a lot of the chromatin accessibility peaks that correlate with gene expression. So it can work very well, and we think that going forward in the future, if we can collect multi-omic data, like generating RNA-seq and ATAC-seq data from the same cells, it will give us even better resolution in identifying, more and more peaks. So technology advancement is really key.
Thank you very much. So the next question, just disappeared. Yes, go ahead.
I have a question for Dr. Zhang. I'm not sure it's on. Can you hear me?
Yeah.
Very interesting. I was just wondering how you're dealing with the levels of activity of a particular cell type, because as you know, they can be, the same cell type can be either silent or very active, moving from silence to bursting activity. So how does the transcriptomics deal with that variability?
Yeah, sorry, I actually didn't hear it very clearly exactly what the question is. Yeah.
Renee, could you try just one more time? Sorry.
Yeah, for Dr. Zhang, I'm wondering how you're dealing when you're studying transcriptomics with variable levels of activity of a particular cell type. So imagine a cell type is either silent or very active. Is it going to come up as different cells in your clustering analysis?
Yeah, that's a great question too. It gets into the issue of distinguishing different cell types versus different cell states. And we actually don't have much knowledge about that yet, because so far the single-cell transcriptomics data mainly have been collected in a baseline level, but it's an extremely important question you're asking, and as we collect more and more data of cells at different states, we want to see whether they segregate into different clusters or they are just like a continuum of the same cluster. We can also use immediate early genes, the expression of immediate early genes to differentiate cells that are silent or active. That's a great question. We need to collect more data to really understand the answer.
Great, thank you. Here's a question for you, Dr. Fair. It says, one of the issues in fMRI is that the tasks used, even in ABCD, are very basic reward or cognitive tasks, and then the hope is that measures from these will somehow predict or associate with complex clinical symptoms and behaviors, such as in neuropsychiatric disorders. Is there a need to generate better, more sensitive tasks that tap into complex cognitive processes and also multiple tasks related to specific functions to improve predictive power? And this was from Rajita Sinha.
It's a very good question. It's a very and a difficult one. It's not very difficult to answer. The answer is yes. Of course, that improving on the paradigms to what will assist with the overall goals. The difficult part, of course, in something this massive is the early choices that have to be made to generalize and to be able to be as maximally helpful to all the different types of questions that are trying to be asked. So there was a lot of hand-wringing about the choices, but for that specific question, I do think there's probably better tasks that can be developed or should be over time.
Thank you very much. Please.
Hi, the question is for Dr. Zhang. Great talk. When you are trying to characterize single cell types in areas of the brain that have never been characterized before, like for instance the human hippocampus, how do you work with the fact that certain canonical genes have been known to be expressed in, let's say, the cortex only in cells like that belong maybe to vasculature or oligodendrocytes, genes that are, for instance, related to immaturity? And how do you understand how they can possibly be related to other type of cells in a region like the hippocampus that's, for instance, in mice has a proliferating niche, and in human we don't know yet. We went through this kind of problem when we were trying to combine our data to characterize the phenotype of certain cells in these regions that have never been analyzed before. So I wonder how you would approach this problem.
Yeah, So that's a lot of questions about cell types. Yeah. That's a very interesting question too. I think, as we collect more data, I think we first, as I said, we want to have a complete characterization as much as possible, generating a reference atlas that's covering as many cell types as possible so that you can map your cells into the taxonomy. But we also beginning to realize that there are quite a bit of immature cells, both neurons and non-neuronal cells in different parts of the brain as well. And a natural extension of the next stage of work is to characterize cell types in the same way during development and having as many dense time points as possible so that we can link the stages of the different cells across time. And then that will facilitate mapping as well. So really the cell type distribution is a combination, as I said, continuity and discreteness. Discreteness more related to identity of the cell, whereas the continuity could be more related to the state change of the cell, either during behavior or during development. We need all that information. Yeah.
Thank you. So I had another question for you, Dr. Boas. It was asked whether the question disappeared from my screen, but oh, here it is. It came up again. One of the speakers wanted to know if NIRS affects mitochondrial respiration.
So this is low-level light therapy. So this is a field, photobiomodulation, that's been around for a long, long time, and recently just becoming more carefully controlled studies are being done in humans showing that effects. So basically the idea is that light absorbed by cytochrome oxidase can enhance the efficiency of oxidative metabolism and it has some impact. So, and near-infrared spectroscopy, of course, is using the same wavelengths but lower powers than people are using in those therapeutic approaches. So there's certainly similarity, but also differences.
Great, thank you very much. Dr. Kepschel, will BARC, can you map projections from a particular type of neuron, such as a neuron expressing a particular receptor in a specific brain region?
So not on the level of a viral infection, as you would usually do with a crate-dependent virus, if you're using AAV, because the virus that we're using for all of the barcoding is an RNA virus, so it recombinases out. With BASIC in particular, you can now do the cell type assessment post hoc by looking at the genes that those cells are expressing. So you map all of them and then find out which cell types they are afterwards.
Great, thank you. I'm going to take one last virtual question and then invite you to come up and talk with the speakers. So this is for both Damien and David. There's a striking contrast between needs hundreds or even thousands for a reliable brain signature for blood flow associated signals like fMRI, pushing rapidly to the level of individual with wearing devices for blood flow signals FNIRS. Could the two presenters comment on this?
Sure. Yeah, so I think the important piece of this is that the number of participants that are required to identify a reliable signal is kind of dependent on what the question that you're asking. So if the question that you're asking is what parts of the brain respond to this specific stimulus or something like that, then the ends can be actually very low and you can get really high signal. If the question is, how does that same stimulus differ as a function of executive function or a disorder or something where the effect sizes are going to be much smaller, then you need lots more data to get an effective result. So hopefully that helps answer the question, but yeah.
I was thinking the same thing as I was watching Dr. Fair's presentation. I was worried, oh my goodness, this question's going to come up after my talk. But his response, I agree with completely. It depends on the question being addressed. And actually, the discovery you have from fMRI can then help you identify those questions that could be addressed with small n.
Thank you very much. So I want to thank everyone who attended, both virtually and in person today. Anyone that's here in the audience that would like to ask questions, I know we ran out of time. Please, our speakers would love to talk with you now or later throughout the meeting today. So thank you very much. We conclude the plenary session.